know, we are here to celebrate Giorgio Parisi. So I wrote a few lines of most of the personal recollections, and then I will read a message coming from the Minister of Research. So Giorgio Parisi has been a member of this academy called Accademia of Sciences detta since the year 2000. And among the members in the history of the Academy who were awarded the Nobel Prize is number 8 and 4th among those that obtained it in physics. The other two being Guglielmo Marconi in 1909, Enrico Fermi in 1934, and Carlo Luca in 1984. So the interval is time interval, so you support in any case, it's with enormous pleasure and gratitude that the Academy is gathering here to celebrate him with this small conference. Giorgio has been a student at the University of Roma, now called Scienze, as I was, but maybe because he's 1 year older than I am, or because he graduated in physics while I graduated in mathematics. I have no recollection of him as a student. I must say that at that time, mathematics, especially algebra and number theory, which is more especially algebra, was very much Dualist. And we felt as a point of honor to ignore physics altogether. This, of course, changed over the following years, maybe starting with the seminal period of human license, which was published in 1972, and was called Myth Opportunities. And the title tells a lot about the situation at that time. And then the change continued with the developments of conformative theory, string theory, and so on. So that in a way, nowadays, we could say that mathematics is applied physics. This was a joke. Okay, coming back to Giorgio, after my graduation in 1971, I left for the UK and came back in 1975. Then the first time I heard about him was in Salerno in 1976 from the Japanese physicist Ken Yoshida, who unfortunately passed away a few years ago. Ken was coming from Rome, and all the young people in the common room were eagerly asking, so what is Super Giorgio doing? Then they explained me that Super Giorgio was indeed a super talented young guy, whose surname was Parisi. After that, I happened to work in the same university as Giorgio, first in Torlonia at the beginning of the 80s, and then starting in 1996 at La Sapienza, from which both we eventually retired. Still, and not because of the stupid so-called superiority complex I mentioned before, we never really interacted. I remember that with my colleague Claudio Fucesi, who I always consider my mentor, we organized a couple of gatherings in 1909, and Giorgio explained us his ideas to us. I confess I understood very little, and things like that. Okay. Still, I developed in the years a very great admiration for Giorgio, for his energy and open-mindedness, which I completely miss, as he appears to be able to, on the one hand, to produce great results, and on the other, be a public figure with great curiosity and insight to travel diverse matters. At this point, I owe some thanks. So, it is a duty for me to thank the people and the institutions who helped us to organize this conference. First of all, my sincere thanks go to the staff of the Accademia, and especially to the leader, Giulia Trimani. They followed every aspect of the organization with great efficiency and dedication. Second, as you can see, the conference is taking place in the beautiful Teatro of Villa Torlonia. This is the first time that the Academy of the Accademia is hosted in this theater, and my sincere thanks go to the Sovrintendenza dei Beni Culturali di Roma for having given us this opportunity, and to the Teatro di Roma for the organizational and technical collaboration. So the theater is very good, probably not so perfect for a scientific conference, but still, this is the first time we shall decide here. This conference owes its existence to the dedication and hard work of the two organizers. On my right, Lidia Berghina, and on my left, Riccardo Tegna, who are both members of the Accademia, and that I most heartily thank, as I believe we all do. Finally, Professor Cristina Messa, the Minister of Research, was supposed to come to the conference, but at the very last minute, she had to cancel their presence in person. However, she has sent a message asking if I could read it. This is what I'm going to do now. The message of Professor Cristina Messa. In 2021, Shugulo Manabe, Klaus Hasselmann, and Giorgio Parisi were awarded the Nobel Prize in Physics for groundbreaking contributions to our understanding of complex physical systems. That is a sign that the study of complex systems is of paramount importance. Complex systems are a field of research that is now acquiring the characteristic features of a well-formed area of science, with its own object, conceptual apparatus, and methods of analysis. Furthermore, the transdisciplinary study of complex systems offers innovative solutions to outstanding scientific questions and global societal challenges, including scientists from very different areas and backgrounds. Thus, bridging the natural and social sciences, enriching both and reducing the gap between science, engineering, and policy. It enhances long-term harmony between science and societal needs. Today we celebrate Giorgio Parisi, but also the renewed position of the Italian science, research and researchers in the international. The Italian government recognized the future of national research and higher education by devoting one of the 6 missions of the National Recovery Funds to education and research, Mission 4, where the support of the researchers throughout his career, together with other actions, is a building block towards the mission approach. The implementation of the National Recovery Plan foresees both reforms and investments. In terms of reform, I'd like to mention a few that are specifically conceived to modernize the university system at large, such as the reform of university degree rules, which is in line with the needs for transdisciplinarity as put forward by the study of complex systems. The reform indeed aims at updating of the present method used for constructing the didactic orders of the university degrees courses, reducing the existing rigid boundaries which severely limit the possibility of creating cross-disciplinary individuals. The reform of PhD programs, which aims at updating the regulation, simplifying the procedures for the enrollment of candidates, researchers, national and international, in PhD programs, and strengthening those measures dedicated to the building of programs that are not aimed at academic career. And lastly, the reform to foster mobility and simplification has made Italy an attractive destination for researchers. This intervention also includes support tools of research mobility from academic enterprises, increasing the collaboration between public and private organizations. As said, the National Recovery Plan foresees both reforms and investments. Here again, I'd like to say a few. We're investing 500 million euros to increase scholarships for university access and numbers, funding and benefits. I'm glad to recall that a share of the investment is reserved to female students in STEM disciplines. I don't know, do you say STEM? I don't know.
We are investing more than 1 billion euros for PhD programs. Following the aforementioned reform of PhD, on April 11th, a ministerial decree was published to launch the award of 7,500 PhD positions in different fields, including curiosity-driven research. And as we talk about the important research activities managed independently by young researchers, I'm glad to announce that we are investing 600 million euros to provide young researchers with immediate first experience of research responsibility. The action aims at attracting young researchers who are beneficiaries of high-profile international grants. As I said, the government is highly committed towards the scientific community, and decides or better to complement the PNRR with long-term structuring actions. Our ministry has increased the national investment in fundamental and applied research. Again, I'd like to quickly mention the FIAS, Italian Fund for Science, for the ERC-like projects presented by early-stage and advanced researchers. In 2021, we received more than 2,000 applications in response to the very first call. This year, we have 150 million allocated budget. A second new national tool is FISA, Italian Fund for Applied Science, inspired by EIC, European Innovation Council. It will fund projects presented by researchers in collaboration with industry. The first call is now open, and with an allocated budget of 50 million euros in 2022. Lastly, I'd like to mention the PRIN, we usually call it PRIN, Research Projects of Significant National Interest. This is a well-known instrument for funding 3-year projects to promote curiosity-driven research activities, both fundamentals and oriented. Here we have been able to increase the budget up to 749 million euros, thanks to the synergy of national 204 million funded by FIRST and the EU funds, 545 million funded by PNRR. In conclusion, let me underline once again that curiosity-driven research is one of the most noble human being resources. It always existed and will always be the deepest part of human development and knowledge. So I think we could thank the minister. So now I think we are almost ready to start, but we said we would start at 40, I think, right? So maybe we should have a 3 minutes. I'm a fast guy, so I mean, and so we should have a 3 minutes break and then start in 3 minutes. Okay, but in any case, we are going to prepare maybe the slides of Giorgio Parisi. I don't know. Where is Giorgio? Parisi is around. He is there. So, the speaker is here. Do we have to? Are we ready for the talk? Yeah. So Giorgio, come. Non lo so, forse. Dove vuoi stare? Lì o seduto? Come vuoi? No, qui va bene. Ok. Questo. Ok, so. Forse bisogna pulire questi microfoni. Ok. Ok.
Grazie.
Okay, so let me introduce the first speaker, Giorgio Parisi, that is known to everybody for obvious reasons. And then he will talk about the complex systems from a physicist viewpoint. Okay, you have 40 minutes. Thank you.
Okay, thank you. Thank you. So let me first say something that I do not like those people which define themselves a complex, expert in complexity, because I think that there's a very nice quotation from a book that says that all simple systems are similar. Every complex system is complex in its own way. Therefore, I think it was Tolstoy or something like that, but anyhow, this is something important, because the point that maybe what is important is some kind of attitude to look into the system. For example, one point that was stressed by Kuhn in his book on scientific revolution is that the scientific revolution is a change of glasses that you use to look to the world. And so it's clear that there's a change of glasses that people that looks with a glass of complexity sees different things that others do not see. But the point is that after the system must be studied in its reality, and one could not import things from other fields. The other things that first I will give you as a disclaimer, that I started to work, to be involved in this business of complexity just by chance. And the things happened in the following way that is quite strange. I was interested for some reason to polymer, to the radius of polymer in solutions, but polymer, not only linear polymer that have branched points that are polyfunctional junction. So they have many bifurcation and so on. And however, I was not really interested in polymer, but was interested in the mathematical tools that the people were using in polymer, because I wanted to use for another problem of high energy physics. So in '78, I did the first work with my French friends on the problem on field theories, high energy physics. So I was looking, we see that this was related to this polymer, so I started to look to the books on polymer, to the paper on polymer, on Physical Review and other reviews, what was known about the statistical mechanics of this branched polymer. And at that moment, from the paper, I believe that is Lubinsky, but I'm not sure about the author, that was saying that people were used the replica method for studying polymer, and there was something like a footnote that the replica method was used for other people by Sherrington and Kirkpatrick, let's say, for studying spin glasses, and however, the results were inconsistent. And it was like a warning. Now, it's clear that you see that if someone presents you a tool to differentiate functions, and it tells you, however, some function gives the wrong result, it's clearly that you may have some suspicion that the method, you would like to understand what's going on. And the second thing, I was interested that, for a lot of time, my idea was that theoretical physics need a more precise tool. So I was interested in all possible new tools that one could use. The normalization group was a great idea that came out in the 70s. And also I considered the fact that that there is a tool that in certain cases you know that it does not work, but you don't know why. It's something like a shame. It's a shame for the category. We have also, as theoretical physicists, we have a certain pride And it's clear that it was considered it was a shame. And so I started to study the model. My first conviction was, just reading the quotation, the first paper of scientific impact, that there should be some more or less trivial mistake. After I found other papers that was explaining which was the mistake was essentially that people were evaluating some integral, assuming that the function was a maximum in some point, but that was not true. There was a technical problem, but it was not clear at all how one could adjust to fix the problem. So I started to work on the problem, and after a certain amount of time, and not too much, a few 6 months, maybe less than 6 months, I was able to find essentially the first how to go in the direction to fix. And after 1 year, I think it was completely fixed. However, the problem that was, I was not really not interested at all. This Sherrington-Kirkpatrick model was done for the physics of glasses, of spin glasses, but I was not at all interested to the species of spin glasses. It was just for me, it was just a mathematical model. Just like a computation integral, something more difficult than the computation integral that have to be done and find the right way to compute the integral. However, at the end there was something strange, that the technique that was used were something that do not make sense from the mathematical viewpoint. It's something that many people do not understand, how you could write down mathematical formula that do not make sense from mathematical viewpoint. Now, the answer is simple. Suppose now that you want to compute the perimeter of a square. You have a very simple formula that as function of the perimeter of the square, as function, you take a circle. You put a square inside the circle, and you want to compute the perimeter of the square as function of the radius of the circle. Now, you know the simplest thing is to take the radius of the circle equal to 1, and you get some formula for the perimeter, which I do not remember what it is. Now, you can do for 3 squares, for a triangle, do with a pentagon, you can do with an hexagon. And after, it does not need to be a smart mathematician to write a formula for all number of sides. It's something that contains a simple formula which contains a sign and so on. But it's just a standard thing. Now, suppose that you look to this formula and you say, well, I want to use this formula that is well defined to compute the perimeter of a polygon. It will contain 1.5 sides. Now, it's clear that when you start to, of course, you can write the formula and you get the perimeter of the formula, but this does not make sense because a polygon should have an integer number of sides. A non-integer number of sides is something that does not make sense. And also the formula is quite strange. For example, you can say what happens if the perimeter, if the number of sides is equal to, for example, 2/3, you find that the perimeter is negative, which also does not make sense. So this type of nonsensical mathematical, writing the nonsensical mathematical is very easy to construct. You take formulas that are valid in one domain, or a theorem that are valid with a certain number of hypotheses, and you use in another domain, or you use in the case where the hypothesis, the theorem are no more valid. Now, the things was done in this nonsensical way, and the formula were very nice, but the physical meaning of the formula was completely unclear. Now, after this takes a few years, after thinking to the problem, and not only by myself, but also other people started to work, it was discovered that these formula were related to the fact that the system has many equilibrium states. Now, let me just come back to the idea of many. Therefore, the main idea that was put in that formula, but I did not know about that, is that the system has many equilibrium states. But now let me give you an historical idea what many equilibrium state means. Let me start with something that should be familiar to most of you, evolution. Now, you had the idea that evolution is gradual, that is the gradual idea of evolution, that you accumulate small mutations one after the other, and that is certainly correct. But the thing that you may ask, not only about the genotype, but you can ask about the phenotype. If you look to the phenotype level, there is the idea that was put by Eldredge and Gould of punctuated equilibria. That you have, as far as the phenotype is concerned, you have a long period of stasis in which you accumulate mutation, but the phenotype does not change too much, with very fast, rapid punctuation period of evolutionary change. So therefore you get a situation where you don't see a gradual change, but you change a nearly constant, a very fast change, a nearly constant situation, and very fast change. And the idea is that at a certain moment what happens that the type of mutations that you may have accumulated just arrive to the threshold that they give a change, a big change in the phenotype. However, this is more or less, maybe Gould will not agree with this kind of presentation, but therefore the idea is the following, quite clear, that you have a system like a living object that has a situation in which you have a situation of equilibrium where the system in this kind of species remains stable, and it goes from one situation of equilibrium to another situation of equilibrium in a very fast way. Now, this is something that is not typical only of species evolution, because you can think you can just look to geological era. To what happened in geology. We know that the herbs as a macro system may stay in some period, the Devonian, Cretaceous, so on and so on. And usually you see that there is an equilibrium from the if you look to the geological rest, to the rocks and so on, you see that there is an equilibrium, and at a certain moment this equilibrium is broken. It may be broken by an exogenous event like a meteorite, or it can be broken by some broken of equilibrium. The equilibrium is no more stable, and you go from one situation to another one. And therefore, you know very well that you can clearly see for a type of rocks, for a type of mineral, which period it was looking to the remain. And therefore, the other situation in which you have the idea of multiple equilibria is the theory, for example, that was done by associative memory, by how the memory works. It was a theory that started with Hebbian. After it was go on, there was a great contribution by Hoffield. After it was a contribution of Amit. And essentially, the idea of associative memory is, of memory in some also that the system of neurons makes neurons change state very fast. The type of a spike of neurons is ordered a millisecond. Of course, if you want to, however, to decide if the neurons is active or non-active, you have a bigger time scale, because typically legs between the spikes of the neuron are 40 milliseconds, something like that. But anyhow, it's a quite small time. And it's clear that we may remember, I mean, not remember, we may think to a given recollection, to a given object for a much longer time than the microscopic state. So this means that our memory is in long has the possibility to remain in this equilibrium, I mean, state for a long time, and after it can jump very fast to another equilibrium state. And the fact that there is an extraordinary number of things that we do remember means also that our part of the cortex, which is responsible for memory, may have an incredible large number of equilibrium states. And you can go on, for example, the other things that you can look to some animal. The behavior of each animal or living object may stay in many different states. For example, a dog may sleep, you make a small noise, the dogs jump. So you have equilibrium state. By equilibrium state, it means maybe equilibrium state is not the good way to call, but it's what people usually say, a state that is more or less constant, at least in some part of it, in some appearance from the phenotype viewpoint, for much longer time than the microscopic scale in which it changes. I mean, the memory, for example, speaking of memory, what happens is that when you recall something, your neurons some of your neurons are activated. Activated means they have a typical, as I told you, 25 spikes per second. But of course, the memory is going to change, because in a moment there's one neuron that is spiking and another neuron that is spiking. So if you look at the microscopic level, just millisecond per millisecond, you see a system which is in rapid evolution, a rapid change. If you look average of a half a second or also 1/10 of a second, you say that you can say some neurons are activated and some neurons are not activated. And for example, there were very nice experiments that were done for monkeys that you can see that this pattern, they have convinced monkey with giving some amount of juice to think, to remember some kind of object they presented. Typically, they presented a picture, and after 10 seconds, they present a second picture, and the monkey have to press a button to say it is the same or it is a different one. And if the answer is correct, the monkey got a juice, get something in his mouth that he likes. So when doing these things, people can do some electrodes in the brain of the monkey and see that the signal of certain neurons remain constant. I mean, constant in the sense average. I mean, the neurons remains active for a long time, for all the 10 seconds. After the monkey press the button, the signal disappears because the monkey has no more need to remember the stuff. So there was, however, when I started, and this is, and therefore I would say that the main essence, the first essence of complexity is some kind of system which have a multiple equilibria. That it can remain, I mean, it's quite clear that if you want to have a bigger repertoire of behavior, the repertoire of behavior is very large, and the fact, because, for example, suppose that you take the water that in this bottle, in the bottle over there, I mean, it cannot do many things. I mean, now it's liquid. Now, if you heat, it may become vapor, it may become gas. If you frozen, it may become ice. But there is not the behavior of water is just very simple. Normally stays in one single macroscopic state. You have just only one phenotype at the moment, and it may jump to another phenotype, to gas, solid, or some. Of course, there are a few different types of gases and so on. But I mean, not so much about. If you take something that is complex, the situation, the richness of the behavior is something that is very, very important. However, when I was started to do these things, I was not at all aware of this complex stuff. So it was a surprise that these things that the formula that are written using this strange idea of polygon object with a non-integer number, or sets with a non-integer number element, was related in some way to this type of this type of problems. After we went on during our studies, and we discovered that we could do some kind of statistical. I mean, what we were doing was some kind of statistical mechanics, a way to formalize the statistical mechanics of systems that have many equilibrium states. And now, and also the things that we discovered that was something quite amazing. That if you take this system like spin glasses, now maybe it's better that I take out a mystery because people have no idea what spin glasses are. So I think that I should give some explanation. Okay, spin glass are some kind of material, but what was interesting, not only the material, it's interesting the mathematical model. And I think, as Russell was pointed out, mathematics is a science which does not know what it's speaking about, something like that. I mean, that is quite clear. 2 plus 2 is 4, but the 2 may be 2 child, plus 2 child, maybe 2 apple, maybe 2 elephant. So once that you pose the problem on a mathematical framework, you lose the essence of you lose what we call the substance of the problem, but we have only the mathematical relations. So instead of speaking about the spin glasses, and the problem, for example, computing the ground state of spin glasses. And I could think to present a different problem which have the same mathematical formulation. And the different problem is something like that. Suppose that you are inviting at your house 12 people, and you have 2 tables by 6, and you want to assign a table to these people. Now, it's clearly that if you are inviting Garby with someone else, it's clear there are some people that you cannot put in the table with Garby. that is quite clear. So you may just put start to write. This guy can be with this one. This guy cannot be with that one, and so on. But it's clearly that this problem is something that, of course, the best situation where you can find a solution where all the desires are expected. But it's clearly that cannot be done in many cases. For example, if you have 2 people, and each of these 2 people do not want to stay in the same table as the other 2, as far as you have 2 tables, you cannot, of course, having these things. Now you have a more complex situation. For example, I want to stay with John. And I also want to stay with Frank, but John and Frank do not want to stay together. It's clearly that my desire to stay with both of them is not respected, or John and Frank may stay in a different situation. Now, it's clearly that is a problem of you may have two different versions, that you may just say friend and not friend, or you may just assign a numerical value to the friend and say I'm friendliness and so on. But now it's quite simple. You can just look to the possible solutions, to the possible disposition. I'm not taking care at which place of the table they sit, just design the table. And there is something like, I can discuss how many there are. Well, the 12 divided 6 factorial square, but anyhow, the number you can compute easily a possible way. But the things that happens, you find which is the best solution, or the 2, 3, 4 best solutions. And to that solution, you can write on a piece of paper how many desires have been satisfied, and how many desires have not been satisfied. I mean, it's something that you usually don't do, but you can do. Now, suppose that the problem becomes different, that instead of having a table by 6, you have a table by 600. Of course, if there are 600 people on the table, you usually don't care because the table is larger. But suppose that you do care, but now it's just the mathematical model, and now you want to study this model in the limit where the number of people, the number of places on the table is going to become infinite. Now, and you want to find in principle, you want to know the properties of the best configuration, the best way that you can assign. Therefore, of course, for each particular instance, you can compute find the best way, but the thing that you want to give a theorem, the efficacy, the satisfaction, the best way in the limit n go to infinity is given by that number that you write in the formula, you compute the number with 10, 20 decimal places. That is what essentially I was doing. And now the things that was quite surprising in this case, that not only that there were many nearly optimal or nearly optimal solutions, were essentially there were many these optimal, near optimal solutions, but the thing that was quite surprising is that you can organize in a taxonomy way. I think that not only the biologists, but also the non-biologists know what the taxonomy is. It just is the Linnean division of living objects in different taxa, that you can have a kingdom, after kingdom you can have philia, for philia you can have classes, order, family, genus, species, subspecies, and so on and so on. So you have this kind of hierarchical classification, which is a typical classification that we do not only for living objects, but many, many times if we want to classify objects and so on, our brain tends to classify objects in a hierarchical way. The thing that was quite interesting and was completely surprised that if you look to this problem, of sitting people on tables, the solutions, the nearly optimal configuration can also be classified in a hierarchical way, with many, many levels, just in the same way that in the biological history. But we know in biology why it was, because in biology we know that this is an effect of the history. The phylogenic sorry, the phenotype differences reflect in most of the case the genotype difference. The genotype difference reflects the type evolution. So these three, because essentially what I speak of something like a genealogical tree is a tree which is deeply rooted in the history. It's not a tree that, on the other hand, when we have this kind of system, spin glasses or this type of optimization problem, we have this kind of, it appears in some way, doing the computation and so on, you discover that the state have also this hierarchical distribution. And of course, the things from a mathematical viewpoint is not so simple, because if you have to describe a tree which have infinite branches, and the branches are all type of level, the description is not easy, and unfortunately there have been a few great mathematicians. One was Nobel, which gave a construction of this tree, mathematical construction of the tree, of a continuous tree. There's another thing that was done by Bolthausen with other technique. Anyhow, therefore, this is the way that when we realized that there was this taxonomic situation in the case of this model of spin glasses, I started to understand that all things were related to complexity. And indeed, the point was essentially the following. I mean, it's something like, if you want, it's something like putting your baggage in the back of your car. I mean, you try in one way and you nearly put everything. Now, if you want to improve the things, quite often you don't have to move things by a small amount. You have to look for a completely different solution, and you try again, you change something basic, and you find something that is completely different, and maybe after 2-3 trials you find the right one. So, okay, so therefore this in some sense explains the fact that, that the problem that you have essentially is a problem where you have some kind of request that are difficult to put together. You have one baggage that is flat, one baggage that is round, one baggage that looks like a shoes, it is a shoes maybe, and the request of this object is difficult to put all together. So you have some kind of requests that are not compatible, because of course you are not filling the baggage of 100%. And that, and therefore, this type of complexity is something that may come out when you have a system we have incompatible requests. You cannot satisfy everything, and you have to find a compromise. But as you know, there may be many, many different types of compromise. So this was essentially a starting point. After this starting point, it's clearly what happened that there was a lot of interest on this type of ideas in many other systems. As Phil Anderson, a famous Nobel Prize, put that spin glasses are a cornucopia of ideas that have been used in many, many other fields. And for example, they've been used for optimization problem. They have been used by amateur good friend Polinsky to understand the memory. They have been used in many other ideas of complex systems. For example, the idea that Ricardo was using ideas that were developed in this framework to understand the protein folding. There's a lot of possible biological application. And also, if we want, what is important also is a metaphor. I think that the science has one importance of some discipline or the other, in not only by technical tools, but also on metaphor. Therefore, this idea of complexity is bringing some metaphor to other science in order to go on. So that I think that I can. How much should I leave?
5 minutes, more or less.
Okay. Okay. So there is. There is a lot of things that, for example, people can ask. For example, well, there's some of these things do not have a nice, a very nice answer. But for example, one thing that you can ask is the following: suppose that you have an island. Well, no island is really an island because you have always. From the, I mean, suppose that you have a same system which is close to immigration. I mean, not human immigration, species immigration. Therefore, you have the number of species that is on an island that is at the beginning is limited. And suppose that we do not look to the system for such a long time that you have a speciation and so on, but suppose, therefore, if you can ask how many types of different species you may have on an island, on a closed system, with a given area, because you have a given. Now it's clear that model system is large, that you have some ecology. So you can ask how, for example, the number of species in an island depends on the surface of the island, neglecting immigration, or the other things that, for example, was something that was asked by, for example, by other people was, for example, you can look to different type of cell. We know that each metazoa, each animal, has many types of different cells. But of course, the point is that if you take a simple animal, they just have a few types of cells. If you have a large, more complex animal, the type of cell is much more differentiated. Now, the thing is now just because the information of type of cell it depends on the genome, you may say that if the genome is very short, extremely short, you can just have one type of cell or a few types of cell. If the genome is very long, you may have many types of cells. Now people were trying to ask the questions, in at least in simplified model, how the number of different cells, phenotype of cells that you may have, it depends on the length of the genome. Of course, the question is clearly that the question is ill-posed. It is completely ill-posed because the length of the genome, the length of the genome does not mean that the information contained in the genome is not directly proportional to the length of the genome. Because, for example, we have many plants, I think lilacs or other plants, we have incredible huge genome, which just comes from the fact that the same chromosome is more or less replicated many times, so this is not important. And also, but I mean, this is something just giving some kind of ideas or questions that you may ask. The other question that you may ask, that is important, is, for example, what is the properties of a random heteropolymer? I mean, we all know that the proteins are some kind of a polymer. Essentially, a protein is a long polymer of maybe a few hundred amino acids, typically, and it's a heterogeneous polymer because it's not long, just the same sequence. It's just, it's not arginine, but and it is more, not really random, but it is there's a lot of diversity. Now, the problem that you may ask, from this point of view, what happens to a random heteropolymer? Now the question is, from the viewpoint of biologists, biology may object that proteins are not random. The sequence of a protein is not a random sequence because it has been selected by billions of years of evolution. On the other hand, what you can ask about what was happening before evolution and when evolution was starting. Because when evolution was starting, it's clear that the evolution had not yet selected the proteins and does not have selected the DNA, RNA or what else. And you can ask, therefore, which were the building bricks from which evolution has started, which are properties of the building bricks. And also you can ask, just make the comparison between a random heteropolymer and a protein. You can understand which are the properties of proteins that have been selected by evolution and which are the proteins, properties of proteins that are present in spite of evolution. And therefore you can try to understand, because it's clear that you may have a different, evolution it just only makes change things that is needed. For example, for example, there have been many questions that have been asked. Come si dice capicciolo in inglese? Nipple. Why men do have a nipple? It's clear that these nipples are not used by men. So you may ask why evolution puts men with nipples. The answer is very simple. Nipples are very useful to female, and to having males so different from female that they do not have nipple would have cost you a strong work of evolution, and it's neutral. So the fact that men have a nipple, do not have a nipple, is a neutral property, and evolution does not care to optimize the neutral properties. So there's a lot of systems of this type. So I think that I gave some idea of the type of idea that I have to put in this game of complexity. So I thank you very much for the attention.
Are there any questions from the audience? I can give the microphone.
Okay.
Yeah.
There is different equilibrium state. Okay, so let's say the healthy state is an equilibrium state. The cancer state is an equilibrium state. So what we're doing as a medical physicist, what we're trying to do is to find something about the kinetic of movement from one state to the other. Now, do you think that the kinetic is important to understand how the cancer state can stay, if you like, or can just disappear for some random effect? Is a parameter, is a phenotype that we must continue to study?
The kinetics is certainly very important because it's clear that it tells you which amount of things you can explore. And it's something that what really happens with the system, real system, it only depends on the type of kinetics. Of course, the point is that also the kinetics, from the theoretical point of view, is much more difficult to understand than that the equilibrium point. Because you see, because the equilibrium point, take this as a metaphor of fitness. The equilibrium point has some local minimum fitness. You have something like a fitness landscape, and the species stays in a local point where the fitness is optimized. Of course, this does not make sense because you cannot speak of a fitness of given species and so on, but that's just giving me a very simplified. Now, it's clear that in order to go from one phenotype to one species to another phenotype, you have to go in some region where the system is not optimized, the fitness is lower. So this may happen in the dynamics in which you can cross this barrier to go to some fitness, to some local minimum of fitness, to another local minimum of fitness, is extremely important if you want to control the type of system. So you have, for example, you may have allopathic evolution, you may have a different kind of evolution, you may have a different mechanisms like exaptations, but it's clear that, but also if you want to understand from just from formula the dynamics, also for simple dynamics, this is much more difficult to understand for the equilibrium, because you have really to understand the barrier that goes from one stable situation to another stable situation. So it's clear that in evolution you may have some unfavorable mutation, which after a few generations gave another mutation which is more favorable, and the combination of the two is better than the original one. But this is the dynamics, this is very difficult to understand in general.
So you seem to be suggesting that in early evolution there were already polymers. You also earlier you talked about hierarchical organizations. So in my mind, but I'm not sure what it is so that evolution started with oligomers, dimers, trimers. They were useful, and then they connected with each other and were selected for because they were already useful. What is the information? Which of the two is it?
I'm not really an expert of evolution, but I was using evolution as a metaphor in order to explain some of the concepts that arise in these things here. And of course, a real understanding what evolution was going on is clear that it's a complete and open problem, and for the moment I've never written anything on real evolution. So for me it was just a way to present this idea, a possible application to try to understand this. I mean, it's the same idea that the mathematics is maybe okay.
Thanks, Professor. Very interesting. You mentioned about evolution and complexity. And as a biologist and ecologist, I'm fascinated by complexity, of course. And in ecological systems, all complex systems have a higher stability. This was a concept was promoted by Darwin and then developed over time. So my question is, in your remarkable experience, you have also the same evidence from physical systems different from biology. So the more complex they are, the more stable they are, and they're buffered response to forcings and to other factors, or?
No, I don't think. Well, I don't know. I mean, the answer is not. I mean, it's not clear to me. This is not.
I have a question, if I can. In one of your old papers, you were talking about the propensity to learn of models of neural network based on, say, spin-glass theory, in a sense. So nowadays, we are kind of observing in practice neural system, artificial neural system, that appear to have quite a propensity to learn. And so this also might be connected to also what you were saying before about hierarchical organization. So it seems that things are coming together. So do you have an opinion of when, you know, kind of artificial neural network, biological neural network, and physics are going to, to meet really?
Well, what happens in the future is very difficult to understand. I mean, as was stressed also yesterday by Mark Mezzar, we see that artificial intelligence and the system that works very well with artificial intelligence, that is deep learning, the deep neural network, is something that we know that works, but we do not have a complete understanding of why it works. And also deep neural networks are something that are completely different from the way that this type of neural network works.
I think, unfortunately, I have to close the discussion because of time. So let's thank the speaker again. Our next speaker is Lilia Alberghina, who will speak from the desk, and she will entertain us on system metabolomics to investigate the complexity of metabolism as driver of cancer.
Thank you very much. But probably of course, you might believe me that I'm a little bit stressed to talk after Giorgio Parisi, because, of course, after his inspiring talk, what is my aim is to bring you the interest for the importance of the beautiful complexity of living organism. If I may have my slides, please. And what is really important? Is important the fact that understanding the laws of biological complexity probably are going to give us some important clues on problems that so far we are not able to face, of great interest for scientific and social aspects. So let me have the possibility to give you an indication of the numbers of the complexity. So when we talk complexity of a living being, what are we saying? But we are saying that thanks to the great improvement of the DNA revolution, the so-called molecular biology period of the life sciences, we are now in the condition to be able to recognize practically all the molecules that are present in a living being, and not only to recognize the structure, but to know and to measure the quantity of each molecule. For instance, the messenger RNA is not... Go back, please. Ok, questo non funziona. For instance, to understand, to measure and to determine how many different messenger RNA are present in a cell. It's something between 2,000 and 3,000 messenger RNA are normally expressed in a cell, and changing the condition, you can have that a given messenger that you can see in the lines is expressed red or is not expressed very much green. So we are in condition to measure and to characterize the molecule. Not only we are able to go down at the structural level, down to the, let's say, atomic level with the present knowledge. And so is it possible to have the structure even of a big complex like the ribosomes. And the number of this complex, for instance, the ribosomes is in the order of millions for each cell. So the quantity, the structure, and the specificity of these molecules is extremely high. What can we do with these numbers? And this when the complexity starts. Can we just use statistical analysis to determine the correlation? Of course, this is the easiest things to be done. Next slide, please. But it is clear that the next slide, please. Since 1948, Werner Weber wrote that paper that I think is very interesting for people that are interested in complexity, in which he was saying that we have two types of complexity. The disorganized complexity, like the molecule in a vessel, of a gas in a vessel, or the organized complexity. And the organized complexity is the complexity of different molecules in a cell, for instance, so in a living organism. In order to understand the biological complexity, we need to have a system approach. And the system approach requires that we try to make predictive models of the different molecules or organization of the cell in order to understand the reasons why the cells behave in a different way, both at a macro scale, or for instance at the cell level, or at a molecular scale if we are able to go down at this level. The reason is that what we are interested in, the function, why a cell divides, for instance, that is the typical property of a living organism, is not dependent on a specific molecule, but requires the interaction or even a large numbers of molecules, and appears of an emergent property. So something that given by the interaction, not only by the simple addition of the different molecules. This kind of study, as I show you in a moment, gave origin to the systems biology at the beginning of this century. And here you have a book in which the first results of systems biology were presented. That has been done with Hans Westerhoff, that is going to be the next speaker. So, in order to understand the complexity, biological complexity, organized complexity, we need the interaction of experimental findings and molecular modeling. What has been able to do systems biology in these 20 years. Systems biology started from theoretical biology and integrated its studies with the molecular structure of the recombinant DNA technologies, of the data of what we might call the biological revolution of molecular biology, and starting with the year 2000, as I was saying before, we start to talk about systems biology. What has been done? Models of short pathways, models of specific reactions, models of constrained models of metabolism, even a model of an entire cell, but is a small bacterial cell of about 500 gene products. So we don't have the molecular model of a human being, and I think that this is probably far in the future and probably would be not very important. What we need is to have models of specific important function, going from the cell cycle of yeast up to the interaction of brain and mind, what is the basis of the mind activity. It is expected that in these years, in the next future, we are going to put together the modeling aspect and the experimental data coming from the post-genomic analysis, so to have predictive models of complex biological functions. So something that requires hundreds of thousands of molecular models, and if we are going to be able to understand this kind of process, there is the real possibility that both the scientific understanding and the practical applications would be very important for society. What is a system? Because using the words probably is important to define what is something that we are talking. We call a system a number of interacting parts, molecules or set of molecules or cells that behave generating function in a process that requires both the molecular structure of the nodes, but is very sensitive to the interaction between the two. So changing the force of interaction may change the function. As I said before, they come as emergent properties. The strength is extremely important in determining the network function, and this is one of the reasons why sometimes the function of a system is difficult to be predicted. It's not because we don't understand what is the network of the nodes that determine the system, but is changing the strength of the interactions. There is an input-output response, often non-linear, and this makes us, out of a possibility of a predict. The causality of the events may be circular, so something that we think is following a cause instead is at the beginning of the series of events that generated that cause. Stochastic events that generally may perturb a system are quenched by presence of anti-chaos mechanism. There is a memory of the past. There is a strong dependence from the environment, and these biological systems are able of auto-replication. These generate a number of features that so far are difficult to be fully understood. So, what is interesting is that it's possible to identify a module which turns to be the appropriate representation of the system. So when we consider a system, we often look to both to the general laws that determine the activity of the system, as well as what is going on in the module that determines the function. Next. We started looking at a module of a cancer cell. Cancer, of course, is enormously, a lot of different features, abolished the immune response, as well as does not depend on the general factors for the proliferation, is resistant to apoptosis. So there is a number of so-called hallmarks of the different cancers that we know. But there is one small property that we might consider as a module for all the different if we understand this, probably we are going to be close to understand all the others. It's at the basis of all the other activities. And this is the ability to grow in vitro in condition of density when the normal cells are not able to grow. Under these conditions, the messenger RNA are altered. There is about 1,500 messengers that are stimulated and about 500 that are downregulated in a given cell line. What is interesting is the fact that the messengers that are stimulated, if we compare human and mouse response, appear to be about 300 in common between mouse and human. And this 300, it was very exciting to find out that they have mitochondrial localization. So it appears that alteration, a profound alteration of mitochondria does take place in cancer cells. In fact, the mitochondria are much less active than in cancer, the black, as compared to the normal cells. And if we test in vitro the biological activity of the cancer cells, at that time, there was year 2010 or something like that, there was not all the literature that has been collected later on, and it was the first time that we were able to see that the change on metabolism in cancer cells requires glutamine to be used as a source of both carbon and nitrogen in a TCA cycle, entering as alpha-ketoglutarate and going both clockwise and counterclockwise. The cycle normally goes clockwise, coming from pyruvate and acetyl-CoA. Instead, in this condition, it does not come from glucose and pyruvate, at least in part, but comes from glutamine down to alpha-ketoglutarate, and from ketoglutarate goes counterclockwise to isocitrate and then acetyl-CoA. So what is seen here is that the metabolism of cancer cells is able to use glutamine both as a source of nitrogen and as a source of carbon for the synthesis of biomass. This appears to be a very important change, and in a way brings us to the source of that basic module that I told you we in general look after when we are doing the systems biology analysis. And changed also the way we think about the importance of the genetic information in the cell, because it's clear that we have a great importance of the genome, or also the epigenome also. So the genetic information is relevant, but what the cell does not depend only from the genomic information, but from the environment. The environment in terms of nutrients, the environment in terms of modifiers, both external and internal. And this is the change of paradigm that the systems biology brings. Is not only the genetic information responsible of the activity of the cell, and therefore at the basis of disease and so on, but is also important, the environment. How do they express? They express through the interactive metabolic pathway that making different building blocks, the ATP, sustain the three possible fates of a cell: growth, death, or differentiation. All three of these fates of the cell require a different assortment of protein, a different assortment of the biosynthetic activity, and therefore a different metabolism. So, as Jens Nielsen was saying, any perturbation of cellular physiology have a metabolic fingerprinting, so a specific pathway, and is the change of this part of the metabolism that is going to be responsible for the final activity that we are going to measure. Therefore, we need to understand the rules that control the metabolism and need to construct mathematical models. If we talk about, the complexity of a cell, we have billions of bases in a human genome. We have millions of components of different molecular, different protein, for instance. Metabolism is interesting because starting from yeast down to human, metabolism is sustained by about 10,000 different enzymes. And the network of metabolism is substantially conserved during evolution. So instead of millions of 100,000 or thousands of different components, we are down to 10,000, which is much smaller than the other numbers. But it is a much smaller, quite complicated, because the map of metabolism is complex. Metabolism has a different possibility of going one way or the other, and therefore we need to develop new, interesting mathematical models. And this is, in a way, the challenge that I am bringing to you and to all the colleagues that are interested in complex systems, because I think they are finding the way of making sense out of the different changes of metabolism is going to be very relevant for the understanding of diseases. And I start to tell you what kind of models we can do and what so far the scientific community has been able to find out. Models can be as a network that are very nice, they can have thousands and thousands of nodes, but they're static. And so to understand something very active, very movement like metabolism is a nonsense. It would be extremely nice to have a dynamic model, but the dynamic model that are based on mechanisms, so could be based on the mechanism of the metabolism, require parameter estimation, require knowledge on the properties of the enzyme that normally are not feasible. So what we have available now are the constraint-based models. So this is, you can see, is really the map of the metabolism, so the 10,000 reactions I told you. What can be done is to fix the amount of nutrients that enter in this metabolism and to determine by some way, for instance, as a level of messenger expressed in a given cells, what is going to be probably the level of the enzyme, because if I find that in a cell glucose 6-phosphate dehydrogenase is not expressed, I can cancel that reaction and have a network that behaves in a given way. The work has been done, and for instance, for the condition that I told you before, so the ability to use glutamine, the ability to use a lot of glucose, some sort of low oxygen available, the cancer cell behave in this way. So this does not work really. Glutamine enters, like I told you before, so enters the level alpha-ketoglutarate. What is interesting is that from the Krebs cycle, the red arrow goes to glycolysis, to PEP, and then to lactate. So this means that you can make fermentation of lactate coming from glutamine, which is an unusual pathway. Not only can go under these conditions, but also it is found that the same pathway can be done in a different way using a different redox reaction. And altogether, it seems that these are the rules that govern the metabolism of cancer cells. So there is an increasing utilization of glucose and glutamine, low oxygen availability. Glutamine is used as a nitrogen and carbon source. The TCA cycle is branched. This is extremely unusual. In fact, they didn't want to use the word and the title of my paper because nobody has seen before. The glucose oxidation goes to lactate, and a large number of redox reactions sustain the metabolism. This condition yields the maximal growth rate, and this is central for a cancer behavior because the attack that the cancer cell goes to the host is to grow in the host and therefore to alter, to damage the functioning of the host. So cutting down the ability of cancer cells to grow would allow to contain the disease. What is, after that, both the technologies allowed to study metabolism and the results on cancer made metabolism an interesting topic. These same technologies have been used to study different diseases. And the strange, interesting thing is that practically all common diseases have a different metabolism. I'm going to almost to close. So here we are at the question, and the question of systems biology and complexity. It is clear that it would be extremely interesting to be able to understand the laws that govern metabolism in cancer and in most the diseases. And this would open complete different pathways for the possibility of treating important diseases. There are all things that scare us, from cancer to neurodegeneration to things that we would like to control, like the wellness. Why the diet don't function? They don't function because they are not able to control the pathway. Okay. At this time of the knowledge, it is we try to organize the ideas of what to do in order to start with the data on metabolism on different cancer conditions, for instance. And it is clear that it is not easy to try to reach to have the design principle of the metabolism in different conditions. This is due to the fact that the dynamic of cancer growth depends upon the oncogene, but not only, also depends from the tissue in which the oncogene is expressed. A RAS tumor in the lung is not the same thing as a RAS tumor in the breast. And therefore, as I'll show you in a moment, does not make sense to consider only the oncogene as the target to be hit by the new drug. The sensitivity to inhibitors, therefore, depends on the interaction between the oncogene and the differentiated state of the cell, which we generally don't know. But metabolism is the marker of this difference. There is the hope that machine learning will allow to find the rules of the metabolism altered in cancer, in different kinds of cancer cells. Why we think it is important? Because the only other things that is done in this period is the use of genetic markers here to in a experiment done on patients that have been treated and that have been marked with a different, for instance, these are the old patients. They are screened for genetic markers. On this basis, you can say that the black ones are sensitive to A drug and the other one are sensitive to B drug. You can go down in the red area, see, they're sensitive to A and B, and then the other to A and C. And in this case, the target has been the DNA, excuse me, but a DNA alteration of the gene, the DNA repair alteration. It is clear that the DNA markers are not satisfactory. Look, this is the only clinical data, and the difference between the non-screened with genomic markers and the one screened with genomic markers is only a few days, a few weeks. So the genomic is not enough to give indication, specific indications on how to treat efficiently patients. If we gain only 2 weeks, is not something that is worthwhile. Therefore, by the end of my talk, what next? We have to find the way to get the same kind of prediction, but focusing on metabolism, not only on genes, on the mutation of genes, in order to be able to apply to the patients the best treatment possible. So the end of the basic molecular biology, in a way, is here, is at the end. What we need is to understand the complexity in order to treat complex conditions, which goes from the environment to the disease, goes from the ideas of the environment to goes down to the specific conditions. So with that, I finish, and I thank you for your attention.
Okay, we are a bit late on schedule, so we have time for one question, if there is any.
Thank you. So from this perspective, Professor Alberghina, Professoressa Alberghina, scusi mi.
What's that?
So at present, people are, when tumors are found in human beings, they are excised and there is a sample taken for transcriptomics. Would you recommend also to do metabolomics, or is it good enough then to only do metabolomics on a tumor sample?
No, I think that it's difficult at the moment to visualize what to do, because of course you cannot to get how to personalize at the level of patient. The only possibility is that experiment of metabolomics will be done on the treatment that comes from surgery. But this is something that is at a given time, and maybe the patient has problems later on, with years after a treatment with a drug. So this would make a real problem on making the specificity. My idea is that if we go to the redox control of metabolism, it would be possible to recognize few specific cluster of metabolism. For each one, it would be possible to find new drugs, because redox control is not at the moment a target for specific drugs. So the specificity would come probably much easier if studying the dependence of cancer metabolism from metabolism, we will be able to find new rules that controls the tumor growth. At the moment, it would be not simple, the idea to determine the metabolism along the several years in which a patient needs to have specified drugs. So it's the future, is not the present time. But I continue to say it's not enough to have the genetic markers. The genetic markers allow to have a small improvement, but the small improvement is not completely satisfactory in terms of the patient.
Thank you very much.
Thank you again to Olivia. And now we have to rush because there are too many talks, but I think we can wait for a couple of minutes, and the next talk is by Hans Westerhoff from Amsterdam, and he will talk about intelligent artificial intelligence for health, from new thermodynamics to a fundamental different approach to network diseases. Thank you very much.
Thank you very much, Mr. Chairman, and thank you, Professor Ressa Alberghina, for this marvelous presentation. It will be interesting for me to talk about this, but it would be difficult. So my talk is different from this. It actually carries...
You are perfectly right, but I don't know who is taking care. They are doing this.
So this is a bit of a challenge. So, can I have the first slide of my presentation, which starts with the nice picture of the symposium. Right. Okay. So we are full of hope. Then, the next best thing is, of course, to start with the questions of my presentation, but that may also be difficult. Okay, yeah, that's it. That moved a bit too quick. Lee? So it is indeed a great honor to be in this theater of the Villa Torlonia at the symposium in honor of Professor Parisi, and it was also a delight to listen to his talk. You will notice a strong difference between the talk of the biologist and the physicist. Physicists are so intelligent, they don't need any slides because they remember everything by heart. Biologists are so terrified by complexity that they can't remember what they're talking about, and they need slides to show you. So that's what I'm going to use. But indeed, it has been marvelous until now. I will talk about intelligent artificial intelligence for human health. But I'll go all the way from thermodynamics, from physics, therefore, through chemistry, and then to biology, and a bit to medicine, I hope. So that will be fun. And in fact, I will start very fundamentally what science is, and then I will discuss 4 cases, probably don't have time for 4, but where information has guided artificial intelligence, and then I may end with a perspective. So, science addresses causation, that is increased likelihood, and therefore I think that corresponds to the purpose of science, that is actually to predict what will happen, that's one, and the second is to understand causes of what happens. And the first of the two is also done by artificial intelligence, the second is not. For that we need something else. And I find that important, other people also find that important, causation. But what is a cause in terms of physics? Or biology for that matter. Now, I will address that by using thermodynamics, non-equilibrium thermodynamics. There will be more talks later in the afternoon, I think, on non-equilibrium thermodynamics. So I'm looking forward to this. Now, this is actually a standard picture that you may remember. This is about entropy. And you may remember that there may be a system that consists of a volume with a septum in between. There are 4 particles. You may not have had this precise example, but it's something similar. And the 4 particles may be on the left, all of them on the left, or all of them may be on the right. Or they may be sort of randomly, some on the left, some on the right. Now, what we would, as humans, say that the upper 2 are ordered systems, and the lower, 14, I think, are chaotic systems. Now, all of these systems fundamentally have the same probability of happening, so that means that we will usually find a chaotic system, and more rarely we'll find the ordered system. And we will find that if we started with the ordered system, the system will appear to move towards the chaotic system. And this is causation. That's it. Nothing more in big systems. You may think there is something in causation like energy. Energy may cause systems to change. And that's what you see here. This is our system. It is in high energy. It has lots of glucose. This can be degraded to lactate. If you allow that to happen, no glucose imported, this might be happening. This would be happening spontaneously. Why is that? Well, it's downhill energy, but energy is a conserved property, so the energy has to be exported to the environment. This will be done in terms of energy quanta, and it will end up in the environment in a random way, random arranged quanta of energy. This is called heat. You will see that the environment heats up a little bit. So basically, because this energy is now in many, many quanta that are random, that still we have the same system. We move from a system with a low probability, where there was high energy, this was not yet there, to a system with a high probability, where there is low energy here, but many, many quanta are there. So it's actually the same principle of causation, that you move towards higher probability. That is the cause in biology and in physics and in chemistry. It increases, systems move towards higher probability. And of course this has been called entropy. Entropy, high entropy is high chaos, high probability. That's the same thing. Okay, now there is a history behind that by another Nobel laureate who actually wanted to be a Nobel laureate in physics, and I'm sure Giorgio will appreciate this story. This was Ilya Prigozhin. And my good friend Jörg Stucki once stood in an elevator with him, and he said, "Professor, you must be very satisfied that you won the Nobel Prize." And then Ilya Prigozhin said, "No, I'm not satisfied, because it was the Nobel Prize for chemistry only." I wanted it for physics. So Giorgio Parisi should be satisfied, of course. But Prigogine developed this scheme. Of course, this picture, basically, you have a system. The system can be changed. There's a coordinate of change. You may increase its volume, or you may increase its concentration of ATP. And we're plotting here the entropy, the probability, the sort of thing I've been discussing before, as a function of this system coordinate of change. And I've just told you that causation, the cause is towards higher probability. That is what cause is. So the system will want to move towards higher probability. And the only way it can do this is actually by moving its coordinate to the right-hand side. So you will see this now happening. The system will move until it's at the top. And you'll remember this, systems at equilibrium have maximum entropy or minimum energy, where you mean Gibbs free energy. So that is understood thereby as an effect of causation, which has to do with probability. Now, Giorgio also mentioned briefly fluctuations, and I'm going to address that because that's important. Although the system here settles in stable states, maybe not all the time. And that is this issue about fluctuations. Systems may change away from where they are. Now, what is this? It seems odd. Actually, it isn't odd, because it is the same scheme here. We have realized that we only have 14 cases that are chaotic, but still, or as many as 14 cases that are chaotic, but 2 of them are ordered, and they're all equally probable. So you would expect that every now and then the system is in this state, because this has the same probability as any one of these. So every now and then the system will move from this situation to that situation. And that is what we call a fluctuation. And then deterministic behavior is the average behavior that is given by this arrow towards the highest probability. So the system is basically testing every now and then to go to a state of lower probability. And that's what you see here. So every now and then, rather than staying here, this will move to a somewhat lower probability. And that comes about with the change in S, and the delta, the Greek delta, has to do with change, as you remember. And we can develop this Greek delta in terms of, we have mathematicians here, so you should be telling us this story, not me, in terms of a series, and there is the first order term, delta x times the first derivative of entropy with respect to the spatial coordinate, and the second order term. And this is done because the first order term, that's basically the slope of this curve at this point, is 0 at this point and can be neglected in this area. So you can forget about this. The only thing that then matters is the second order term. And that is what Prigogine and Glansdorf then started to continue to discuss. It's a bit confusing, this 2 here, but that's what it is. So this is called the delta 2, superscript 2, that comes, agrees with this. And now what Prigogine then showed, Glansdorf and Prigogine showed, is that this delta 2s is always negative. But you can see this, of course, because we are at the top, say you're up the top of the Mont Blanc, and you want to move about by fluctuation in Europe, you're always going to go down, well, Western Europe, because the Mont Blanc is the highest mountain in Western Europe. So that is what you have. If this is the highest position and you move away from the highest position, it must always be negative. That's what it is. In delta 2s is always negative, but the important thing is you can then show this by statistical mechanics. If you have a solution of substances with chemical energy of the substances, and these are the mu, the chemical potential, amount of energy has to do with the concentration that is then in here. And it's basically the derivative of this with respect to number of particles of any sort multiplied by this. And you may recognize that, you may remember or maybe not, that this is actually the difference, the differential of the Gibbs energy. Mu is the differential of the Gibbs energy with respect to Ni. This gives you a positive matrix, basically. So since this is always positive, then this is always negative. That's what Glansdorf and Prigogine showed. So that's for sure. This is always negative. Now, that reminded him, Prigogine came from Moscow, and Lyapunov was probably also in Moscow. Actually, you will probably know whether he was from Moscow. But he had, before this, developed the stability theorem, which I found always a bit very difficult, magic. But actually it is quite obvious. And it has to do with the same argument. So if we again have this system coordinate of change, and you have any function of the system, that's important and amazing. It can be any function of the system. If you can find any function of the system with the property that is always positive, and that as a function of time it decreases, then you know that there is some function that as a function of time decreases, and that will of course end up here. Because that's what will happen, it will continue to decrease. But this means that the system will return always to this state, even though it's one function. There may be many complex properties of the system. This is now just one of them. It's sufficient that that function returns to that state. And that was the statement of Lyapunov. And so basically his statement was, if you can find any such function, the system is stable. So that's what Prigogine used. Transfer frame Prigogine used, realizing that it's not quite the same, this increase in entropy, but it's inverted. So here we look at something that is positive always, with the entropy, the axis entropy. You saw it was a parabola that has a positive maximum. So basically it's the minus delta 2s that was proposed by Prigogine and Klansdorff to be this Lyapunov function. So now you understand that story. It makes actually sense if you think about this. If you can find any function that it always decreases in time and is always positive, it will end up in its minimum, and that's then the stable state. So minus delta 2s would be that Lyapunov function. And then they verified whether indeed that would, the derivative of, with respect to time of that function would always be negative. And that becomes a little bit more complicated, although you can see the equation is fairly simple. But I have to explain what these two terms mean. Let's think there is only one reaction in the volume. And you have a chemical reaction rate Jr, it's a flux, and this is the fluctuation in that chemical reaction rate. And this is the affinity of the Donder, for instance, but it's actually equal to the Gibbs energy of reaction, and this may fluctuate. So that's all it is, but it looks, of course, complicated. Now, they could actually show that the time derivative of this entropy term is equal to the sum of all the fluctuations in the affinity multiplied by the fluctuation in the reaction rate. And you can figure that out, so there's not going to be any change in entropy if there's not a process doing it. So those are all the processes. And they found that there would be this expression. Now let's forget about the sigma, just to simplify. So we have this, and this is the fluctuation in AR, so it's actually the change in AR from the standard state. This is the change in JR from the standard state. You do the trivial multiplication of this, you get this expression. And you may say, okay, what does that help me? Well, actually helps you a lot in equilibrium, because in equilibrium, A is 0 and the J is 0. So all these three terms disappear. You're left with this, and that's a total entropy production in the system, production of chaos in the system. And the second law of thermodynamics tells you that entropy is always produced. When you're in equilibrium, nothing happens, no entropy produced. When you're away from equilibrium, entropy is always produced. So that's just the second law of thermodynamics telling you that this term, this term is positive. So if this term is positive, then this is negative. That's the story. Well, sorry that became a bit complex. But you can see that Klansdorff and Prigogine then could show that close to equilibrium, systems are always stable, which you would reckon they are. But you know, mathematicians and physicists need a lot of language to tell us things we already know. But it's good, because now we know for sure. That was again a joke, sorry. Not intended. You had a joke yourself, which you didn't laugh about. So now, near equilibrium, therefore, delta 2s is such a Lyapunov function. They also found that before I told you this is always true, So then they realized that they would only have to show, which they had now done near equilibrium, they only would have to show that this is always negative, also away from equilibrium, and then this would be a stability criterion. Then the system would be stable. So that's what they said. They were inspired to say, well, if this is always larger than zero, then we have this as a general stability criterion. And that was a fantastic story, and one of the reasons why Prigogine got his Nobel Prize. But then this became problematic. It's not quite this simple, because you're away from equilibrium, and these terms are not zero. And what are they? And then with long thinking, for instance, by Joe Kaiser and by myself and by Lonsdorff, we realized really that this depends on the kinetics of the system. And I'm very pleased that question was also asked to Giorgio Parisi, whether the kinetics mattered and whether you could deal with that. But his answer was also, then it really gets complex if this depends on the kinetics. So it depends on the kinetics. That's where we stay a long time ago, or some time ago, in a book about this. Kinetics and organization are of the essence for non-equilibrium thermodynamics. And mind you, that then tells you it's of the essence for causation. So we need, away from equilibrium, substantially away from equilibrium at least, kinetics, mechanism, organization. Now I interpret this symposium as to talk about biology, not only about physics. And of course biology, through evolution, has channeled into organization. Physics doesn't always need to have organization. Biology always has organization because it had to survive, had to be fit enough. So let's go a little bit to kinetics and organization and real biology. And that's what I do here by looking at enzyme catalyzed reaction. And I plot here the J versus the flux of the chemical reaction, but now a biochemical reaction, as a function of this affinity. And then you get this type of curve for a Michaelis-Menten equation. So the biochemists among you know this. That's standard biochemistry. You can see that it has a maximum rate. So Vmax, you recognize this. So that's all now. And what you find, for instance, that around equilibrium, there's always sort of, you can look at the slope. And around equilibrium, the plot always looks like this. So the basic means that the flux increases with the affinity, and the slope is positive. Away from equilibrium, for these type of enzyme kinetics, you may be more like here, and the slope is also positive. Now, what does that mean? Well, that's actually interesting, because we were interested in this term. You remember this from the Prigogine story. Fluctuation in the flow, flux multiplied by the fluctuation in the affinity. And since this is now L times delta A, because it's a straight part, you get L times delta A square. L is positive because the slope is positive, and therefore this is positive. Again, satisfying this criterion that the change in time of the excess entropy is negative. Okay. So it seems to work for ordinary biochemistry. But then sure enough, biology is diverse. And if you look at another enzyme, one of the interesting enzymes of glycolysis, phosphofructokinase, there you see, if you plot there the J versus the delta G in the ATP, you find a negative slope. And now things fall apart, because now delta 2S is not a Lyapunov function. So that means that depending on the slope of the flux with respect to the affinity, there's something we call elasticity. You do or do not have this as a Lyapunov function. So that really has now strong limitations. That's what I wanted to get at. It's not that useful, that criterion. Further away from equilibrium, you need to know the kinetics. That is negative in the sense you can't understand things from simple physics, as I tried, but it's also good because it tells you that some enzymes may have the capacity to be regulated to be entirely different from physics. So to basically have something like emergent properties which are of the biological type for which you can select, and then you get proper biological function. Now, I told you we must put in how biology works. We must put in organization and mechanism. And let me give you one example. One of the things I plotted here for the same reactions is the absolute flux versus the entropy production. And for both, you see that that is a monotonically increasing function. And that is very important to keep in mind. That means that to get more flux, you need more entropy production, or you need to lose more, dissipate more free energy. So basically, you need to eat to be fast. Because for this you need energy to go in. And that's what you see, that's universal. Now, the other thing in life, if we talk now about the living part of physics, living organisms, it has learned to have a high entropy production because it must be fast. Life has always competed, first with dead processes, and then later with other life. So all the life that persists is fast. And how can life be fast? Well, this has to be high, and therefore either the affinity, the chemical energy of the chemical reaction must be high, or the flux must be high. You can recognize this, that many living processes in living cells are away from equilibrium. It makes no sense actually to assume that they're close to equilibrium. They're not close to equilibrium. Why is that? Well, evolution asks processes to be far from equilibrium. So that's the first. And the second is, of course, you recognize that evolution has played the trick of developing proteins, catalysts, therefore to make reactions faster, and therefore also the J is much larger than 0. So this is actually high. So we know that now. If for the non-equilibrium thermodynamics, that's the type of thermodynamics I'm now discussing of biology, and I'm here comparing this to equilibrium thermodynamics. In equilibrium thermodynamics, there's no mechanism, no kinetics. Prigogine and Klansdorff tried to do the same thing for non-equilibrium thermodynamics, but I'm stating that's simply impossible. So, if you're away from equilibrium, certainly in living organisms, mechanism and organization matters. Because of Darwin, essentially. Yeah? Selection. So, activity of the processes and their organization matters, and we have to discuss things not in terms of pressure and temperature, but in terms of activities of processes in a new type of non-equilibrium thermodynamics. And that's what I'm proposing here. So the flux J, it's the same chemical reaction through a network now, is a function of all the enzymes, E for enzyme. Enzyme 1, enzyme 2, enzyme 3, enzyme N in a pathway, in a network. Same for the concentration, same for the frequency of an oscillation, and same for the realization of time transient. And I'm sure that many of the authors of this afternoon's talk will like this, because they will discuss oscillations in the cell cycle. Now, just an inspiration, this is equilibrium thermodynamics, and this is maybe a merger between physics, chemistry, and the next slide, biology, and mathematics, because I use Euler's theorem here. And we have here the situation that we have energy, entropy, volume, and number of molecules are extensive properties. They become twice as big if the system is duplicated. And then there is Euler's theorem that tells you that then from this you get this statement. This and the du ds, the derivative of energy with respect to entropy, is this temperature. So basically you get this Gibbs relationship, which is a fundamental thing to define the Gibbs function. Now, this is not of primary interest to you, but this may be. This is the analogy now for biology, where we have written things in terms of the fundamental properties for biology, which are the enzyme activities. And J is a homogeneous function of first order of the enzyme activities. And therefore, that's what you see here, you use Euler's theorem in much the same way. And we find that 1 is the sum of the partial derivatives, log, of the flux with respect to the enzymes. So 1 is the sum of the control coefficient. And some of you will recognize some result of metabolic control analysis, which has been very important, developed by Reinhard Heinrich and Henrik Katcher. And this is a very important property indeed of biology. And this is where we used it. We used this in Trypanosoma brucea, which causes the sleeping sickness. We determined this control coefficient experimentally, indirectly experimentally. I could explain this to you in more words. And then we determined the sum of these control coefficients, and this is indeed 1. So that's indeed precisely this. So this is correct. So the mere question then is, where is the highest control? It's not in the rate limiting step, it's distributed. And the highest control was in the glucose transporter. Is this useful? Yes, it's useful, because we want to kill these bugs. These are causing sleeping sickness. So now there's drug design, used to be versus the other enzyme steps, for instance, phosphofructokinase, but now it's towards the transporter. So that's an example where using this type of non-equilibrium thermodynamics may be useful. I'm going to skip this part, Mr. Chairman, because I'm otherwise running out of time. I'm going to move more to the medical domain. So prediction of disease and therapy from metabolic organization, where I'll be using less of the kinetics and more of the organization. Organization that we know since 2000, when the human genome sequence was determined. So we know the sequence, all the nucleotides of the DNA. Fantastic. It's actually interesting also to see that there is so much development after this. But of course we're puzzled because it's so complex, so many bases, so much DNA. And avant la lettre, Edvard Munch already was shocked by this. So we have this. Could we then think of using artificial intelligence? So we do the sequence, we identify maybe all the genes, we make all the connections, that would be binary connections, 625 million for the human, then we train the network. Okay. But you realize to have a good training of the network with 625 million problems, you need more than 620 million data points, experiments therefore. And we don't have them yet. If we sequence all the humans on the planet, we may, but not now. Yeah? So this is not possible now. So we need something more. And this is something that I call intelligent artificial intelligence, or informed artificial intelligence. You take some of the knowledge about the system. And what would we take here? Well, this is an example of a diagram of a living cell, realizing that there's DNA, there's RNA, there's protein, there's chemical reactions here, there's transport, and we're going to use that. And we're realizing also that the work level of the cell, as illustrated by Professor Ressa Alberghina, is actually at the chemical reaction level and at the transport level, which doesn't mean that this is not important. This is also important because it regulates that. So there's work and there's regulation of the work. There's sort of workers like me, and there's managers like the Professor Ressa, who are in the upper regions. So what we did is add this biological knowledge. So metabolism is done by networks of chemical reactions. Every reaction is catalyzed by a protein. And every protein is encoded by a small gene set. This is actually a beauty of biology for physics and chemistry. It's absolutely fantastically organized. If you want to know about the chemical reaction in a living cell, you nowadays try to find a little bit of the protein, sequence a few amino acids, go to the genome, you have the gene, you overexpress it in E. coli, and you determine what it does. That should be well known to physicists. It's one of the most accessible complex physical systems is biology. So that's what's being used here, right? That information, we will use that now to deal with this complexity. This is a diagram signifying the complexity of the DNA sequence. It's a mess, you could say. But we can then look at segments that we can recognize encode a protein. And then we have the entire biosphere that has been sequenced by now, all the microorganisms, and we can search for homologies. Does this bit of DNA, is it homologous to some bit of DNA in an organism? And then, is it then expressed in that organism, and do we know what the function is? And by and large, for most of the genes, we know then from an organism, we have a human gene, we know it's similar to a gene in E. coli maybe, and in E. coli we know what it does, and because of homology, we then know what it does in the human. So basically for all the arrows, which you can figure out are chemical reactions, we then know what the reaction does. For instance, this converts A to B, this C to E, and we make a complete list of this, and then we make use of another property. Basically a steady state, all that is produced must be consumed, what goes in must go out. So if B is produced, it must also be consumed. Well, there's a B here, so we put that reaction next to that. And if there's a G here, there must be a G elsewhere. Yes, okay, we do that. And you can see what develops is a metabolic map. This is the map, the Angro 2 version was shown by Professor Essa Alberghina, a metabolic map which tells you biochemically, metabolically, if you eat something, how you can get from your food to any compound, any chemical molecule in your cells. So these are chemical molecules in the cells. The dots, the lines are the chemical reactions between them, and the food enters here. Now you can, of course, then worry about, this may be a vital constituent that you need for life. Mostly all of them are vital. You may then consider a blockade of this by a gene deletion or an inhibitor, inborn error of metabolism. You don't have the gene, yeah? So you don't have this. There's flux from here to there, so what you will see is this, that this will accumulate. If you can measure that by metabolomics, you will see there's something wrong. That's called a biomarker. And you also saw, of course, that the vital constituent is no longer made. So we explain disease, because no, this vital constituent is not made. Now, so this mutation is an inborn error of metabolism. The most famous type of inborn errors of metabolism is phenylketonuria. I'm going to discuss that. That's seen because the urine is yellow. There's a lack of brain development in the child, and it's most due to homozygous deletion of a certain gene, PAH, phenylalanine hydroxylase. We did that before. Let me skip this one. Now, if we simulate that, we look at the healthy situation, and this is the production of biomass. Sorry about the technicality of the slide, but this is meant to impress you that we are really looking at the entire network. We're only showing the fluxes through the network, which are the colors. You see that this reaction flows well. That's actually biomass synthesis. So you can see there could be growth in the healthy situation, obviously. But now in PKU, you see if you knock out this gene that encodes two enzymes, then you see there is no growth rate. So we understand this now. But why is it lack of brain development? This is a bit older and classical. Now, the story is why brain development? Why is the child growing, but not the brain of the child growing? Right? So it's not just growth. Well, then we looked at neurotransmitters, adrenaline and dopamine. And we found that also dopamine synthesis is lacking in this mutant. That's new. And that is, of course, very rewarding because I think it is maybe 10 years ago, or no, maybe 12 years ago that we were in Milan when the systems biology was started there. You'll see familiar faces there, familiar for systems biology, but also Rita Levi-Montalcini, whom I really admired. She was one of the Nobel laureates of Italy by her discovery of nerve growth factor. And this afternoon you'll find a talk by Anna Maria Colangelo, who is actually from a laboratory named after Rita Levi-Montalcini. Now, what would be the therapy? Since in PKU, the synthesis of tyrosine from phenylalanine is lacking, there must be insufficient tyrosine for protein synthesis. So, would tyrosine supplementation work? And here you see, yes, according to the model, the growth rate would return and the dopamine would return if you give tyrosine. Now, that is interesting, but also very interesting, because it's nonsense. Actually, experimental nonsense, because tyrosine supplementation does not work for these patients. So what did we then do? We panicked. So it didn't work. We were stuck. And when you're stuck, you often go to physicists or you go to medical doctors, right? So we went to a medical doctor, Van Splonsen in Groningen, and he told us, well, he thinks that aromatic amino acids like phenylalanine and tyrosine crossed into the brain by a single carrier, and there's competition. And the method we use, which was flux balance analysis, doesn't allow standard for competition. So that's what you see here. There's one pathway and another pathway, no interaction between the pathways. So what we then did, and that's novelty, we made an FBA plus, which allows for competition, And sure enough, then, fantastic for us, of course, then if we did that, that only tyrosine addition did not give dopamine flux. But if you would take in the diet less phenylalanine, it would. How does that work? Well, if you have lots of phenylalanine, there's competition for the carrier. So the tyrosine that you give, you may give it, but it can't enter because there's lots of phenylalanine. You have to lower the phenylalanine to get rid of the competition. Now the tyrosine enters. So let me stop here. I mean, that was really a nice success story. Let me stop here and skip that. Sorry, Mr. Chairman, I'm already over time. And Chiara Damiani is not here anyway, so this was a bit a plug for her. But let me tell you that I've been discussing this, which you have noticed. I also have a perspective, basically, at this type of thing, where we use, say, artificial intelligence or modeling type of things to understand human health. If we add more of the pre-known information of biology and medicine, this can become even more powerful. Our method is certainly not the only one. There's many people working in terms of that. If we only can associate their results, that's what I propose, integration of these type of intelligent models like Angro-1, Angro-2, then we would be much better poised to go towards individualized medicine. Thank you, Mr. Chairman. Thank you.
So, we start being a bit over time, so maybe I can allow one question, if you want. So there are no questions, so thank you.
There can be two possibilities. Nobody understood anything.
Yeah, or everybody understood everything.
Right.
Thank you very much.
Okay. Good morning to everybody, and thank you to the organizers for having invited me here. It's a great honor and a pleasure. I will talk about the role of information traveling from single cell to systems made of multiple different cells. And before starting, let me thank also my research group, because without this talented research at the Department of Information Engineering at the University of Padova, the work that I will present wouldn't have been possible. Okay, so let's start defining multi-cell systems. Well, we know that tissues and organs are made of multiple cells of different types. We might wonder also what a cell type is. Traditionally, biologists recognize different types of cells by shape, size, and surface proteins, but nowadays we are able to measure the entire proteome and transcriptome state of a cell. And we know that cells exhibit different expression characteristic proteins that make them specific for specific functions. This picture here is a picture of organoids, so organs that are artificially built in vitro. And different colors correspond to different characteristic proteins. So what we can see immediately here is that there are some patterns in space. And indeed, cells are organized in space and time and able to communicate and orchestrate self-assembly, renewal, response to stimuli. And although there is not a sort of controlling cell, and cells do not have a global view of what is happening, still they are able to organize really complex function like human embryonic development, for example. What is possible nowadays, since 5 years in a more, let's say, consistent way, we have the possibility of measuring transcriptome and also of reading DNA within the cells at single cell levels for thousands of genes and for thousands of cells in parallel. And whereas before it was possible somehow to merge the cells and to measure the average expression of genes, now it's possible to look at each single cell. And of course, new technologies bring also new questions. So we might wonder, how is information processed within a single cell? How do cells communicate with each other? And how do they cooperate to orchestrate self-assembly, and how new properties emerge from the collective cell behavior? And what I will be talking about is the role of cell communication in all this, because cell communication might be a central aspect to answer these questions. So before going in the details of 3 examples that I will use in this talk, let me say that when I say that the information is processed within a single cell, I mean something which was shown also in the previous talk. So basically, when a cell receives some sort of signal, information is processed within a signaling pathway, and this might lead to some action from the cell, and also can the information can reach the DNA, which in turn will activate transcription, for example. But we have also information traveling on the other way, because genes can be transcribed, translated, and can be transferred outside the cell. And indeed, one of the most widely used systems of communication that cells use is based on ligand-receptor interaction. So basically, if we have a group of cells communicating with another group of cells, we can imagine that the sender cells secrete a ligand, which is a protein, that binds to a receptor in another cell, and this activates the signaling pathway I was showing you in the previous slide. Okay, so let's make a first example. The analysis of a minimal gene regulatory network of cell differentiation. Experiments suggest that differentiations happens in different stages, in which we have a cascade of similar steps, each one characterized by the differentiation in 2 cell subpopulations. This was also represented in the 50s by Waddington, who used this idea of a function that drives a cell toward terminal differentiation stages in subsequent steps, like the ball that fall down from the top of a hill to lower valleys. There are many mathematical models, many different mathematical models that can be used consistently with this framework. One of those was used some years ago to describe the differentiation into neurons or glia, and from glia into astrocyte or oligodendrocyte. This model was based on, on differential equations. And what is interesting here is that the minimal cell module, the minimal cell regulatory network that is used in the first and in the second switch is the same. We have one driver gene that promotes the transcription of both gene 2 and gene 3, and gene 2 and 3 mutually inhibit each other. And if you see here, the first and the second stages are regulated by the same module. And then, of course, we have noise that is useful to simulate the biological variability, and that might somehow drive the fate of a cell developing in one direction or the other. So we studied theoretically this model, and we also ran some simulation. A key role, of course, is played by the expression of gene 1. And if the expression of gene 1 is sufficiently high or sufficiently low, the cell belongs to an undifferentiated stage, and the gene 2 and gene 3 are almost equally expressed. Whereas if you lower the expression of gene 1, what happens is that for specific values of the system parameter, then you have a situation in which either gene 2 is overexpressed with respect to gene 3, or gene 3 is overexpressed with respect to gene 2. So let's simulate just the first step. What happens is that if gene 1 lowers the expression, so the cell lowers the expression of gene 1, you have a first step of differentiation in two subpopulations. Here you see two genes, the red one and the blue one, that represent gene 2 and gene 3 at the first switch. And in some simulation, you have a high level of the red gene and the low level of the blue one. In some other simulation, you have exactly the opposite. And then you get to the second step switch. So you have a pattern in time of different differentiation steps. So let's put the cells in the space now, in the two-dimensional lattice. Let's run these simulations. What happens is that you have a completely random pattern of differentiation, because what the model of the single cell switch tells us that it will go in one direction or the other. So here different color represents the two different cell types, let's say, at the first step, so neuron and glia, for example. What we observe in reality is, of course, a temporal pattern of progenitors. This is a picture from a recent paper about neurogenesis. This is the mammalian cortex, a model of mammalian cortex development. This is a picture of the Drosophila optic lobe. So we have different cells expressing different proteins, and it's clear that we have a pattern in space. So the question is, how do patterns emerge? So let's imagine, we might think, of course, that there is some additional mechanisms driving the pattern emergence, and let's imagine cells sensing their neighbor. So there is a kind of feedback mechanism between neighbor cells, which we implemented, imaging a positive feedback among type-specific genes of the same type and a negative feedback among type-specific genes of different type. This is just one simulation. If you run it again you have patterns, but of different shape, but you see that patterns start emerging. And if we imagine to add also an environmental effect, so some effect from the border, for example, of our in silico, let's say, plate, you have patterns that resemble also the environment. Of course, in reality, communication among cells is much more complex than what I show you here in this toy example. And this brings me to the second example, a multi-agent spatio-temporal model of cancer and immune cell communication. So, as we have already seen in previous talk, cancer is characterized by abnormal cell division, accumulation of mutations, dysfunctional apoptosis, and dysfunctional metabolism also, of course. And immune system provides surveillance under possible malignant transformation, but when this system is, when the immune system breaks down, tumor, of course, can develop. we can imagine tumor and tumor environment as an environment of many different types of cells, tumor cells, immune cells, cancer-associated fibroblasts, and other types of cells that are autonomous but interact with each other. And they sense the environment, they sense different molecules. Each type has different possible actions because they are different types of cells. Each type communicates by releasing specific molecules, and each type have different nutrient consumption. So we don't have enough details of these systems to build models based, for example, on differential equations, or it would be really complex from the computational point of view. So we limited our analysis to a system made of interacting cells, interacting agents that act in a stochastic way, release specific signaling molecules and are able to react to specific signaling molecules. And concentration of nutrients is really important to regulate tumor proliferation or survival. And nutrients diffusion is modeled using partial differential equations. So let's look really quickly, but in some details, to what the different agents, the different type of cells might do as a type of actions. So we have cancer cells that duplicate and whose duplication rates depends on nutrients. They can also die by necrosis, by lack of nutrients, or by immune death because they are attacked by immune system cells. And when they die, they release substances that recall immune system cells. The first type of immune system cells we modeled are natural killer that are part of the innate immune system. They are attracted in the domain by, let's say, stressed cancer cells, and they represent a fast immune reaction. If they are in contact with the cancer cell, they try to kill it, and if they win, they die, and they release immunostimulatory molecules in the environment. Then we have antigen-presenting cells, which are called in this way because they are able to process the antigens of the cancer cells and to present them to the T cells that become able to recognize them. T cells are another type of immune system cells, of course. They also initiate inflammation, and inflammation contributes to blood vessels dilatation and delivery of other immune system cells. Then we have T cells that represent an adaptive immune response agents. They are of different types. There are really many different types of T cells. We modeled the CD8 plus T cells and CD4 plus T cells. The first one are the ones that recognize the antigens and kills and are able to kill target cancer cells in a specific way, whereas CD4 plus assist other immune system cells in activation, maturation, and attack. After a certain number of attacks, CD8 plus cell die. The activation of CD8 plus T cells represents a quite strong immune response. So the cells need to receive multiple signals in order to attack cancer cells. Besides the recognition of the antigens here, they also need to receive another signal which is called immune checkpoint. And this is meant to, let's say, avoid autoimmune reaction. And then they need also to receive signals by the CT helper, which are the CD4 plus cells, in order to be activated. So you see that here, for example, cells need multiple information, multiple signals in order to fulfill an action, in order to be activated and to fulfill an action. However, cancer keep mutating. Cancer cells keep mutating, and they might evade the immune system, either because they exploit the immune checkpoint that I was showing you before, so they are able to block the second signal that T cells must receive. They can also lose or acquire new antigens, and they can lose adjuvancity. In practice, they become able to release signaling molecules that repel T cells. Finally, last type of agents, we have the CAFs and the Treg that represent the immunosuppressive environment. CAFs in particular promote tumor growth and angiogenesis. They inhibit immune cell recruitment and in general represent a negative prognosis marker. CD4 plus T cells or Treg agents, as I was saying before, regulate CD8 plus action and promote the recruitment of other CAFs and Treg, especially if the inflammation is long-lasting. Okay, now let's put all these agents together so each of them knows which action it can, let's say, performs and in response to which signals. So let's put them together in a two-dimensional lattice of 350 by 350 cells, for example. And let's start a simulation in which at each time step we update the concentration of the different nutrients, and we let the cells fulfill the action in response to what they feel in the environment. Of course, it's a stochastic process. And then, of course, we update also the communication molecules. So this is what can be considered an output of a single simulation, in which for different time points, we can count the number of cells of different types. We can, for a particular instant in time, depict what happens in space, and also have the concentration of glucose, oxygens, and of signaling molecules. Now the question is, can we reproduce key features of real-case biological scenario? So what we try to do, well, we consider human colorectal cancer tissues because it can be classified in 4 different main types, which are characterized by different immune cells infiltrations, different mutational burden, different metabolic landscape, and different clinical features. And we used the Cancer Genome Atlas data of 379 patients and more than 20,000 genes to inform the parameter of the simulation by estimating the fraction of immune cells, the tumor mutational burden, the presence of immunoinhibitory molecule able to block the immune checkpoint signals, and run different simulations for different types of tumor. And what we get is depicted here as the average, let's say, outcome for the different scenarios. So here you get the average and in shadow color the standard deviation, whereas here you have just a single simulation for 4 different time points. And on the top here, you have the number of simulations in which, let's say, the tumor survive. What we get is that these, let's say, predictions, even if they are not real prediction, are, let's say, consistent with the outcome endpoints of progression-free interval that we observed in this type of patients. Data were also taken from the same source, so the Cancer Genome Atlas. Well, what we would like to do, but is still work in progress, is to use the simulator to better highlight the properties emerging from different settings in the different subtypes of tumor. In particular, we are interested in the effect of nutrient consumption, and we are trying to use some tools that were published in the literature to estimate for different patients the metabolic consumption of the tumor. And as you can see here, this is the CMS3 scenario. And if you increase the consumption of the tumor, of the nutrients that characterize the tumor, or if you diminish the nutrients, you get almost the same results. You have changes both in the size and in the shape of the tumor. Now the other question is, can we use the data, the single cell data, to estimate the communication among cells? So which cells are actually communicating and which molecules are used for this communication? Because this is really important to better understand the true systems. Okay, I'm using again this figure here because there are a number of papers in the literature that use single-cell RNA sequencing data as a proxy of ligand and receptor activity. So basically, you take the single-cell expression data, and if you find that a ligand is highly expressed in a cluster of cells correspond to a type of cell, and the receptor is highly expressed in another type of cells, you might, even if RNA is a proxy of protein activity, you might imagine that some communication is going on. Well, actually, there is a high level of noise in this type of data and also no spatial information. So the proxy is really a proxy, let's say. And our idea was to measure the effect of intracellular signaling to increase the confidence that a detected intercellular communication was actually ongoing. So for the intercellular communication, we did what other groups are also doing. So basically, we take the ligand and receptor expression in different clusters of cells. We assign a score comparing this expression with what you would expect by random, and compute a ligand receptor score as a combination of the respective score. You can use the product, you can use the minimum, some fuzzy minimum. You combine the two scores to somehow weight your belief of interaction taking place between cells. Then, to understand if there was an effect on the receiving cells, what we did was to consider the signaling pathways from KEGG, but also reactome, and the transcriptional regulation databases to link the receptors first to the transcription factors within the pathway, and then to the target genes that these transcription factors might activate. So, first step, we converted the pathways taken from Kagan reactome into directed graph, and then we simplified the graph topology in order to make it tractable from the computational point of view. Then we used the page rank, personalized page rank algorithm to score the ability of different receptors to reach specific transcription factors. And finally, going to the target genes, so the genes that the transcription factor activate, we wanted to measure the evidence of an activation or the evidence of a differential expression with respect to some baseline state. So first we compute a differential expression, and then we use the Fisher exact test to, let's say, measure or to assess how many genes which are target of those transcription factors are actually up or down regulated in the cells. Then we put the two scores together to get a receptor pathway activation score. And I'm going to show you the output for a real case scenario. This is a metastatic melanoma, a human data set with more than 4,000 cells and more than 11,000 genes. The cells were already classified into 7 cell types: endothelial cells, cancer cells, T cells, natural killer, B cells, macrophages, and CAFs. And what you get as output from this method is the name of the ligand of the receptor, the transmitting cluster and the receiving cluster, the score of the intercellular communication and the score of the activation of the pathway. Then you have the name of the pathway, of course, and the list of genes that are supposed to or are assessed to react to the communication. And at this point, you can measure for each pair of cell types, let's say how much communication are going on, how many evidence of ligand receptor pairs you have. In this case, for example, CAFs and macrophages were, at least from our analysis, among the 2 types of cells that were communicating more heavily. And you can also ask to a specific cell type which message is it listening. For example, these are the messages that are listened by CAFs specifically. So no other cells are listening the same message. And it was interesting because the messages were related to downregulation of apoptosis, upregulation of angiogenesis, and many functions that are known to be fulfilled by this type of cells within a tumor environment. So some final thoughts to close. The interaction between different cell types has a major role in controlling and coordinating cellular activities, and maybe we need a new instrument to measure this communication in a more reliable way. Even basic cell communication might influence the pattern in space and time, and cell communication, though, is quite complex, involve many different actors, and it's difficult to be modeled in sufficient details. We can use simulation, both to understand and to highlight some properties, but as I was saying, we need a new instrument besides single cells, which of course can be useful to identify, quantify, and characterize cellular communication. We might need new instrument to reliably measure this crosstalk. So there is still a long way to go, and I'm ready for questions.
So I think there is time for one question.
Yeah, you were mentioning the interesting aspect of metabolism being different in the cells, mentioning glucose, but maybe not intracellular metabolites. I was wondering whether you couldn't do, using your methodology, measure correlations of RNAs for metabolic enzymes with receptors.
Yeah.
Or have you done that? Have you tried that?
We haven't, but I think it's a really interesting point. We were trying to use flux balance analysis to applied on RNA sequencing data to measure the consumption of different metabolites by different subjects. But there were some computational challenges. With single cell data, we were not able to scale. We were using some tools available in the literature. So it's something we would like to do, because, yeah, I think it's really interesting. Thank you.
And so we are now the last speaker of the morning session, who is Rémi Monasson. He will speak about generative and interpretable models for aptamer design and analysis of in vitro sequence selection.
Okay, so first, thank you.
Okay, you are really okay. Yeah.
So I would like first to thank the organizers for the very kind invitation to be here. I would like also to apologize to Giorgio for inflicting him a second talk in a row. That means that even Nobel Prizes may have their downsides, I guess. Okay, so I'm going to talk about data-driven models for in vitro and in vivo selection. So I slightly changed the title of the talk compared to what is on the program, because I thought it would be more adapted to the scope of the meeting. And with two applications on DNA aptamers and also tumor responding T cells. So this is going to be very down-to-earth machine learning. It has nothing to do with complex or, let's say, intelligent artificial intelligence we heard about. So it's really machine learning at the simplest level. And there are some reasons we want to do that, and I will explain this later. Okay, so selection is actually a very fundamental biological process. So selection is acting, so for instance, when you get mutation in germlines, then descendants of an organisms will have slightly different phenotypes, or maybe they will be affected in a detrimental way or sometimes in beneficial way. And selection will actually change their life expectancies, the number of offsprings and so on. And this is what we usually think is at the core of Darwinian evolution. But selection may actually take place in different settings. And I want to show two examples which have nothing to do with reproduction. So one is selection taking place in the immune system. So for instance, in the adaptive immune systems. So here is an example of maturation. So maturation is the process of producing highly fit B cells, which are able to recognize with high affinity pathogens. So this is a process which takes place when we are infected by a virus, for instance. And here you see a cartoon of a germinal center. So the germinal center is the place, I mean, we have many germinal centers which are created upon infection. And here you have a cartoon of what is going on for naive B cells here on the left in this dark zone of the germinal center, which undergo mutation and selection process, so absolute selection against their binding of the pathogens, or let's say some part of pathogens, some peptides which are related to the pathogens, and also relative selection which is controlled by T cell helpers, which actually select only the most fit B cells. And you have different rounds of selections in such a way that very good B cells are produced either for immune response through plasma cells or in terms of memory cells, which have the possibility to be reactivated when the new infection with the same pathogen takes place much longer, at longer times. Okay, so so this is one example of selection which is very important, and we'll see also something which is related to another aspect of the immune system, T-cell response, later in the talk. So another example I would like to show has nothing to do with in vivo selection, but is really based on bioengineering. And here you have a cartoon of SELEX. So SELEX is an acronym which stands for Systematic Evolution of Ligands by Exponential Arrangement. And the principle of SELEX is quite simple. Essentially, you would like to create, for instance, aptamers. They could be DNA or RNA aptamers, so short strands of DNA or RNA, which are able to bind some target, typically one protein of interest. So you start with a library here, which may contain some numbers of different randomly created or produced synthesized aptamers, something like 10 to the 12, 10 to the 15, typically. These aptamers are put in a bulk solution where in presence of many protein targets here, and some of them will bind, other one won't bind, and by washing away what is inside the solution, then you will discard all the unbound sequences, keep the bound sequences that can be released from the target, and through amplification, PCR for instance, then you get more copies of these good ones, and then you can do several rounds of selection here, and finally end up with very good binders, right? So depending on the amount of mutations which are introduced here in the amplification, then you get only pure selection or selection with some amount of mutation, right? And this is very useful for bioengineering. It's at the basis of many directed evolution schemes for engineering better aptamers, and also this can be used also for engineering better proteins in a slightly different setting. Okay, so from a practical point of view, if you look at this kind of selection experiments, what you end up typically is data. And the data here, they can be a set of sequences S that you get, let's say, at a certain round. Say, let's say you have done 4 rounds here. So what you can is take a sample from your solution and then sequence the sequences which are inside. And then you will get a sample here, so at a given round. So here I'm assuming there are DNA sequences, just random DNA sequences here that I wrote. And you will get also the number of counts. So for instance, for the orange sequence, you get 100 counts at first round, 300 round 2, 900 round 3, and so on. And this is typically what you get with, let's say, hundreds or thousands of sequences with the counts at different rounds. And the classical way of interpreting this data is to compute the relative enrichment when you go from round R -1 to round R, for instance. And what you do is that you pick up 2 different columns. So R is equal to 3 here. So I pick the column number 2 and column number 3. And I will compute the ratio of the counts here, which tells me how much this specific sequence has enriched in terms of numbers from one round to the next one. And this is used as a proxy for the fitness of the exponential of the fitness, because it tells you how much, this sequence S is actually increasing in terms of quantity compared to other ones. But I mean, this, let's say, processing of the information has some problems. And the problems, you can see them here. I mean, the counts may be unreliable because there may be many biases. And for instance, in the subsampling, if a sequence is not very much present in the solution, I mean, it has low counts compared to other ones, when you do the subsampling, you will end up with very small numbers. So I can compute the enrichment factors from these very small numbers, but it's totally unreliable. There might be biases in amplification. So I see counts actually increasing, but this is not due to increased fitness, but simply because amplification for some reason is introducing some biases. There might be a lot of sequencing errors also, which are affecting the count numbers. And another problem with this technique is that it gives you information only about the sequences which are present in the data, obviously. And this depends very much on the initial library. So the initial library can be large, as I said, typically 10 to the 15 sequences, but this is nothing compared to what you can produce if you compare the whole size of a sequence space, even of aptamers of length, let's say 40, and I will give you some examples later on. So initially, just when we built this random library at time t equals 0, around 0, this is purely random, highly sparse, and then we are stuck with it, and we can compute enrichment factors only for these initially randomly chosen sequences, which is bad. And then the third aspect, which I think is problematic, is that when we get these numbers, it's nice to know that maybe one sequence is increasing a lot in terms of counts, but the relationship between the fitness, why it's increasing, okay, and the sequence content is not clear at all. You need a model to interpret what part of the sequences actually matter for having a higher fitness compared to another sequence. So all these points here actually are strongly motivating to find other approaches which are based on machine learning. And essentially what we assume here is that there is a function which connects sequence to fitness. This function is unknown, and this is what we would like to extract from the data. So here I plot let's say a conjecture shape of a fitness, which is this black full curve here, on top of the sequence space. And what we have in the data are just a few dots here, these blue dots. These are the dots which are the sequences which are present in the data set. But we would like to be able to reconstruct the full function, even for sequences which are not in the data set. And if we do that, we expect that we will be able to understand what actually shapes this function and what part of the sequences, what motifs in the sequences actually matter for the fitness. So how can we do that in practice? We can use unsupervised methods, and I will show you one method that we like. We like it for some maybe good and bad reasons, but it's a very simple one, which is called restrictive Boltzmann machine, which dates back from the 80s. I will define it in more precision in one slide. But all these unsupervised methods, like for instance, you may be familiar also with more recent unsupervised technique like variational autoencoders, VAE. They essentially rely on the same idea. You take data configuration, and then you try to build representation of this data. And the idea is that this is related to variational autoencoders are related to the idea that the data configuration should be encoded into some meaningful representation. And by meaningful or relevant representation, I mean representation from which you are able actually to reproduce the data or some kind of variance of the data. Okay, so the questions here on top of reconstructing the fitness is how we can understand the key features in the data. So can we have interpretable representation which will tell us what are the determinants of the fitness? And are we able actually to use these models, which can be generative, for instance, to generate new data? You want to do design, we want to design new proteins, new aptamers, which is also something which is very important. And possibly we would like to be able to tune the distribution in order to generate new data with some given features. Okay, so the model we use are this restrictive Boltzmann machine. So they were introduced in the 80s. And they are strongly inspired from statistical physics. And I will come back to this point later on. And so there are very simple neural networks with only two layers. So there is one layer here, which is called the visible layer, where the data are presented. So you can put a sequence here. So it will be a sequence of nucleotides, in our case, for instance. And you have one hidden layer, which is also the representation layer, where the value of the units are unknown, and they have to be computed. So, RBM is actually a graphical model which defines a joint distribution for the sequence V and for the representation vector H. And so the log of the joint distribution is actually the sum of three different terms. One term which is the local potential acting on the visible units. Here I'm considering visible units which can take only 0 or 1 values, but they can be one-hot encoding of nucleotides or amino acid. You have also local potential acting on the hidden units. so which can take real values. So, but these potential are, I will explain what they do, but they essentially are trying to restrict all the real values. You don't want values to be too large or too small in some cases. And you have also what is very important, couplings in between the two layers. So essentially, what we try is to do is that we expect that if we learn these parameters here, then the hidden representation is going to extract statistically meaningful features in the sequence which is on the visible layer. And how do we learn these parameters? We can define the marginal distribution on the visible layer formally as the integral over all possible representation of the joint distribution. And then we can just consider this as a likelihood for the sequence in the data set. And we want to maximize, for instance, the product of all the likelihoods of all the sequences in the data set. So this is done numerically. And then we find the best local potential here on the visible units, on the hidden units, and also the couplings in between, which maximizes these likelihoods. So I want to insist on the flexibility of this machine. And in fact, from the joint distribution, you can define the marginal, but you can do more than that. So you can also define the conditional distribution of the representation given one sequence on the visible layer. And that's very interesting because it tells you what kind of representation you can extract for a given sequence. And then the question is how we can interpret this different representation and the different axes in the representation space. And another conditional probability which is very important is the conditional probability of the sequence given the representation. If we understand what the representation means, then we can define a representation we are interested in, which would correspond hopefully to some biologically interpretable features. And then from that, we can define a conditional probability on the sequence space and design new sequences with desired functions. This is a dream. So it's useful to design data from representation. Okay, so why do we look at RBMs instead of other techniques? So I think they are the simplest generative model implementing this data representation duality I was mentioning here. Any kind of other architecture you may think of will be more complex because it will have intermediate layers. And I think it's competitive with deeper architectures in some interesting situations. First of all, it's not true that all situations correspond to large data sets. We are in the era of big data, but in many applications, we don't have big data. We just have limited data sets, and in particular in the second application I will show you. So with limited data, certainly we don't want to have very complex networks with many parameters to infer. And I think another aspect of these RBMs is that actually they are more powerful than you would expect from such a simple architecture, because we can infer parameters defining the hidden potential, this U of H I was mentioning before. And there are many choices you can do or you can look at, you can consider for confining the value of this H variable here. And in fact, this is very different from what people do usually in deep networks, for instance, in variational autoencoders. In variational autoencoders, you map any kind of data onto independent Gaussians, right? And this is the objective. But so this can be done, but if you want to have a complex distribution on your data and map it onto a set of independent Gaussian, that requires a lot of layers, right, in order to do that. Here, since we map onto something which can be parameterized with rather complicated U of H, there is no need for so many layers. And I think a part of the complexity of VAE is due to this unnatural way of mapping onto something which doesn't make sense, except that it's easy to deal with. Okay. So what kind of choices for the potential? So quadratic potentials are very natural one. And they are very natural one because they are related to models which are well known in statistical physics. And these are the models that Giorgio was talking about, some of the models he was talking about during his talk. So maybe I can skip that. Okay, I will go back. And so if you have a quadratic potential, actually, so U of H is just A squared, then when you formally integrate over the H's, what you get are effective couplings, pairwise coupling between the sites in the visible layer. And this is nothing but usual Ising kind of models, which are very much related to the spin glasses here for effective GIJ here. Okay, but we can look at other potentials which are more rich. So for instance, we can have potential as the blue potential. So the blue potential has a cusp in h equal to 0. So that means that having h equal to 0, as soon as you move away from 0, you will pay a lot because the slope is finite. And this is related to the L1 penalization, L1 penalty that people like in machine learning. That increases sparsity of a representation, and that helps a lot to understand what is going on. And you can have, for instance, the red potential, which is a double well, which will favor either strongly negative or strongly positive H's, so some kind of bimodal representation that will help a lot to clusterize the data. Okay, so you have a lot of flexibility. So another aspect of these machines, which I think is interesting, is that they can be made interpretable if we regularize the weights. And how can we do that? There are different ways to do that. I mean, the simplest way is to improve sparsity, increase sparsity. So here, you see, for instance, as a function of the sparsity penalty, some kind of very classical curve, where you have, for instance, the log likelihood of the training set, so which is very high when you don't penalize at all, you don't regularize, and then but there is a lot of overfitting, and when you penalize and you regularize, you see that the log likelihood of a training set is decreasing. Here you see the one of a test set, which is completely different from a training set as no regularization, which is a lot of overfitting, that it will increase. And as you increase more and more the penalty about the sparsity of the weights, then you see that the log likelihood will decrease, which is normal, but actually it decreases in all the applications we have looked at in a very slow way, which means that there is a nice trade-off between increasing the sparsity of the weights and the loss in terms of performance of a machine. At some point, you can stop and say, okay, I want this machine to have this kind of performance. And if you do that, you see, for instance, that the weights here are much simpler than the weights here at the top. You know, usual cross-validation machine learning would tell you that you have to stop here. But if we go there, we don't lose a lot. And still, we have weights which are much, much simpler. And now, if we see that one hidden unit is activated, is strong, then we can understand where in the sequence are the determinants of this strong activation. So there is a huge gain in interpretability. There are other ways to improve the interpretability I don't have time to discuss. Okay. So I want to show two applications of these techniques. So one is on the analysis of a SELEX experiment for the design of DNA aptamers binding to thrombin. And the data were acquired in this paper by Zhu and collaborators 3 years ago. So thrombin is a very important protein which has a key role in blood coagulation. What thrombin does is that it actually modifies the fibrinogen, which is soluble, into fibrin, and fibrin is actually aggregates and forms some protein networks, which is the basic mechanism for coagulation. So now, in order to change fibrinogen into fibrin, thrombin has to bind to it. And the idea of, if we want to prevent coagulation from taking place, then you can try to design very small DNA aptamers, which will bind on the reactive sites on thrombin. And once they are bound, if they are very strongly bound, thrombin won't be able anymore to bind to fibrinogen. Okay? So it's completely inhibited. And this is exactly what these people did. And they have used SELEX, exactly the technique I've shown you before. They have done several rounds of SELEX. I will come back to that. And we have the data from the paper here at the different rounds. And the second application I will show briefly is an analysis of T-cell receptors population in patients suffering from Okay. Okay, thank you. Okay, so, and this is an issue which was actually, which is deeply related to the previous talk. So when we have tumor cell, the tumor cell can proliferate and tumors, mutation can actually also proliferate along the DNA, along the genome of these cells. And one of the questions that people ask is, would like to better understand is how much this process of mutation and proliferation is controlled or not controlled by the immune system. And that may actually depend very much on many conditions. And there is a specific interest in the pancreatic cancer because this is a cancer which is actually has very bad prognosis and is really thought to be very poorly immunogenic. So that is not controlled by the immune system. So we have some data from our collaborator Vinod Balachandran in New York at MSKKC. And the question we ask is, can we have some kind of signature that there is some immunoediting, that T cells are actually responding and controlling cancer, and how does it depend on whether we look at data coming from short-term survivors and long-term survivors? Okay, and I will come back to that later. Okay, so for the first part, first application, so from the Zhu and collaborators experiments, so what these people have done, they have looked at thrombin here, and they have tried to design, they have succeeded in designing, more precisely, very good binders, which are short DNA aptamers, which are made of 2 loops. Each loop is long as 20 nucleotides, so total is 40 nucleotides. And so they start with a random library of 10 to the 15 random sequences. So 10 to the 15 we have to compare to 4 to the 40, and you see that's nothing. So the random library is actually extremely sparse compared to the total sequence space here you could build, you could imagine for 40 nucleotides long aptamers. Okay, and they are done in a specific way so that there is this black part here, which is fixed and which builds a scaffold. And there are 2 variable parts, 20 nucleotides here, 20 nucleotides here, which creates these loops here, which can bind to 2 exosites, binding sites of thrombin, or don't bind, depending on the sequences. Okay, so you start at round 0 with this library, and then you just do more rounds of selects. So this is at round 5, and at each time, so more or less through amplification, the size of the library is kept constant in the experiment, but only a very small subset, which is just 10 to the 6 sequences, roughly, are taken randomly from the experimental recipient, and then they are sequenced. And these are the data we have available, okay? And if you end up with round 8, you see that some sequences have large counts, and they have very high affinity to thrombin. They are excellent binders with dissociation constant of the orders of a few hundreds of femtomolars, so extremely strong binders. And this is one of the examples here, THAD, which is made of 2 loops here, and both loops are actually binding to 2 exosites, and so it's a very strong binder. Okay, so what we would like to do is a kind of a twofold objective. First of all, if we look at data we have at round 5, can we use this data to predict what is going to happen at the next rounds? Is it possible to predict evolution? So it's a very simple setting for evolution, highly controlled, right? It's not compared to in vivo evolution, it's nothing, but it's a nice test ground here. So can we use this data to predict? And is our model able to say something about the fitness? The objective I mentioned before is the score of our model related to fitness. And we could do also something which is complementary. If we end up with data at the end, they are very good binders, but can we use the model, for instance, that we have at the end in order to find more binders, so to design new binders that couldn't be found in this experiment because they were not present in the initial library, simply, but can we find other binders? Okay, so I will just show you some of the results here. So maybe we can look first at the top right diagram here, which shows actually the histograms of the log likelihood found by the RBM model. So again, we use this model here. We define the likelihood here, and the log likelihood is simply the log here. We maximize the local potential on the units and the couplings here, on the data present at some rounds, for instance, round 5. And we then obtain some histogram for the log likelihoods. And this is what I'm showing here. I'm showing that at round 5, 6, 7, and 8. And what you see here, so the log likelihood is increasing to the right, obviously. So we see a distribution which is changing in terms of shape. Essentially, at round 5, it has 3 different peaks. And we see that the leftmost peak is disappearing. We don't see it anymore at round 6. And if we go to round 8, we see only the rightmost peak, essentially, with some kind of tail here. So we have the impression that the whole thing is shifting to the left, simply by the fact that the peak with low log likelihoods are disappearing. They are decaying very quickly. And in fact, these 3 peaks, I'm not showing all the details, but they correspond to 3 kinds of different two-loop aptamers. There are two-loop aptamers which are very good. They bind to both exercises on the thrombin, and they correspond to the right peak. You have moderately good aptamers which bind one of the 2 exercises, either that one or this one, okay? And they are in the middle peak. And you have the bad one that don't bind anything, okay? And this is the left peak, which is disappearing very quickly with more selection, more rounds of selection. Okay, so it seems that the evolution of a log likelihood with the round number has something to do with thickness, but can we make this connection more precise? So, if we look at infinite population, suppose we have an infinite population, so we can forget about any kind of stochastic effect. If I call QR of S the fraction of the sequences which are equal to S at round R, then there should be a simple connection between fitness and QR minus 1 and QR, which is given by this formula. So if I have a fraction of sequences in my experiments at round R minus 1, which are S here, so Q is a number between 0 and 1, then these sequences S will be, amplified exponentially as a function of their fitness here. And you see in front of a fitness some number here, alpha, which depends on R, which takes into account that actually in the experiment, for some reason I'm not sure I fully understand, the intensity of the washing and some parameters of the experiment were changed from one round to the next one. So we assume that there is a fitness which depends on the sequence, but on top of that, there can be some multiplying factor which depends on the round. And then you normalize, and that defines the fraction of the population which is equal to sequence S at round R, right? So it's just relative expansion compared to the other one. So f of S can divided by that can be actually negative or positive. Okay, so if we look at so that means if I divide QR by QR minus 1, I get the enrichment factor I was mentioning before, and the log of this should be proportional to the fitness. So I don't know the fitness, but I know the log likelihood of my machine. So here we plot the log enrichment factors, so log of QR over QR minus 1, as a function of the log likelihood given by the RBM. And we do that at different, from round 5 to 6, this is blue, 6 to 7, and 7 to 8, you see the different colors. And except on the tail here, for very bad sequences, you see kind of, let's call it nice relation, linear relation. It's not perfect, but it's not so bad, which seems to indicate that the log likelihood is proportional to fitness, because you would expect a linear relation if the log likelihood was the fitness, and we kind of see it here from the experimental data. So another way to see that is that we can think about Fisher fundamental theorem for evolution, which tells you that the change in fitness is proportional to the variance of fitness. The rate of increase of fitness is proportional to the variance of fitness in a population. And we just looked at whether this relation is verified or not. And here you see the change of fitness from one round to the other. Of log likelihood from one round to the other compared to the variance of log likelihood. We have only 3 points because we have 4 rounds, but it's compatible with the existence of a linear relation. So altogether, this result points to the following interpretation, which is very much in the spirit of statistical mechanics. So suppose you start with a Q at round 0, so in the initial library, which is flat with S. It does not depend on S. So it will be, you could say that Q0 of S would be exponential of 0 times f of S, right? kind of infinite temperature, just flat distribution. Now, once you start with this flat distribution, you multiply by this, you see that now at round 1, you would get Q of 1 of S equal to exponential beta 1 of F of S, where beta 1 is simply alpha 0, right? The first coefficient here. Now, if you multiply again to go to round 2, you see that you will sum up the alphas. And at the end, if we look at round R, we would expect that we have some kind of Gibbs distribution where the energy is minus the fitness, and the effective inverse temperature is the sum of the relative pressure of selection we have added at each round. Okay? So the picture we have here with this result seems to be compatible with that. So what can we say about the motifs which actually control this effective energy or this fitness? So if we look at the profile of sequences at round 8, which is the last round of evolution, then we see, for instance, this kind of profiles, and it's well known by people in the field, and that is called a G quadruplex motif. And this is a feature that very strong aptamers should have. So this is the average, let's say, logo on each side, but we can have more precise information by looking at the couplings in the RBM, the weights here, which tells you exactly what the different helium units are looking at in the sequence. Okay? And so, for instance, I'm showing you 3 weights here. So these weights here is actually looking, so first of all, a general feature of all the weights we infer is that they consider the 2 loops are independent, so which means that they essentially do not interact, but they bind to each exosite in an independent way. And you see this weight here is some kind of variation of the G quadruplex on the second part, in the second loop. Here you see some kind of long-range AT-rich motif here, and so on, and different motifs. So I think that can be useful because maybe for biologists, then they have something to look at and try to understand whether these motifs make sense or not, right? But they are clearly identified by the machine as important in order to be able to predict the score of the sequences. Okay, and then the last thing I want to show about this application is how it can be used to do design. So the first attempt we did was very naive. We said, okay, if we look at data at round 8, so the last round of evolution we have, we can infer this landscape of fitness with our machine. That's nice. And then we can just, once we have it, we can just run the machine on the computer and generate new sequences and try to see whether they bind or not to thrombin. The problem with that is that these new sequences are very similar to the sequences that are in the data set, right? There is very little diversity in the generated sequences compared to what is already available in the data. And why is it so? That's something actually which is pretty obvious if we consider the interpretation we had here. Think about some kind of very low temperature, so high beta model in statistical mechanics. Well, I mean, if you sample from this thing, you always get the same kind of ground state or slightly excited states, and there is no diversity, right? Except if you have a huge, a huge space. But here we are talking about 40 nucleotides long sequences, and there are not so much diversity at this effective temperature. So the other approach would be to say, okay, so let's look at the data maybe at earlier rounds and sample from the machine, infer from those data. But the problem with that is that there are still some sequences which are pretty bad here, and we don't want, and even in these sequences here, half of them are actually bad. So we would be maybe dominated by these sequences, so we don't want to do that. So what we did is something which is a little bit baroque. We just look at the data at round 8, but we forget about the counts. Okay, forget about the counts. So you forget about the fact that some sequences are very highly present, and so that you put all the good, they are all good because they are at round 8, but we just try to flatten this landscape. And with this flattened landscape, we can actually have a model which is able to generate diverse sequences. It won't generate extremely good binders as the one present in the dataset, but it generates a lot of binders that we can check experimentally. And this was checked by our American collaborators. I will cite everybody at the end. And there is a very good accuracy about the prediction we can do with this machine. Okay, do I have 5 more minutes or? 1 minute, okay, so I don't have time to explain this thing. Okay, I just want to say that we can apply also this machine or these techniques to other data, which are much more complicated data, where we looked at the, essentially the expansion of TCR, of T-cell receptors in data coming from pancreatic tumors, long-term survivors, and we wanted to see whether this expansion is actually specific in terms of, is it due to the presence of a specific neoantigen which is driving or triggering this expansion, or it is just a random process? But I don't have time to explain this. But I mean, I just want to say that the final picture that we get, and if you want to see more information, you can look at this paper, which is going to appear soon. So the picture is that it seems that there is a big difference between long-term survivors and short-term survivors. By long-term survivors, I mean survivors over 10 years, which is very rare for this cancer. What we see is that using this machine, and we look at the top scores, so the sequences of TCR which are the most likely given the machine, but that do not correspond to the sequences with the top counts. It's very different. They seem to be less diverse, okay, in long-term survivors than in short-term survivors. So it's a very strong indication that in short-term survivors, tumors, the mutation in tumors are proliferating, are not under control by the immune system at all, okay? And the TCR actually are just as diverse as you can imagine from the individual repertoire of this particular patient. While in long-term survivors, it seems that the mutations are really immunoedited and that TCR are multiplicating in order to target the specific neoantigens corresponding to this mutation. And there is less diversity in the population of TCR, which is evolving with the mutation. Okay, sorry about being so. Okay, so I would like to thank first, so people in Paris. So for the first part, Andrea Di Gioacchino, who is a postdoc in the lab and is giving a talk this afternoon and has done a lot on this first part. For the second part, Barbara Bravi, who was a postdoc and is now a lecturer at the Imperial College in London. And also Simona Coco, Jorge Cosio, Thierry Morin, and Alexandra Walczak. And our collaborators in New York, Vinod Balachandran, Ben Greenbaum, and Marta Luksha. And in Arizona State University, Jonah Proshik and Peter Salk. And I take advantage of the last 10 seconds that maybe I don't have yet anymore, but that if you are interested in the second part, we have a small workshop organized in Paris next month at the end of June on innate and adaptive recognition of antigens and neoantigens, using these techniques, but also other techniques and with a lot of experimental results. And if you want to have more information, you can look at the QR codes. And, okay, thank you.
I don't know if there are questions. We have also lunch, so we can... So one question.
Yeah, the last bit was, of course, quite interesting. I assume that whether the long-term survivors and the short-term survivors, you analyzed that before you knew they were long-term or short-term survivors, or was that after?
No,
when did you get?
We knew, but I mean, but we didn't do that. Okay, so it wasn't you mean double-blind thing? No, but we didn't use this information. So. Okay, so in the training of the RBM, right? So if you look at what we have on the yeah, so I was very fast, so I couldn't explain that. But essentially, if you just have to look at the right axis here. So we have the sequence data, TCR sequence data. Okay, so some of the patients are where it's long-term survivors, or the ones who are short-term, right? But then we train the RBM, and that from the sequences, right? There is no way we can cheat with that. The only thing you can do is adjust the penalty parameters for regularization. We do L2 and L1 regularization on the weights, right? That's it. And then this TCR dissimilarity index is that once you have scored with the RBM the 25 top sequences for each patient, then we just compute some kind of mean Hamming distance between the sequences, right? So there is no parameter to be adjusted there.
So thank you very much, Remy, again for the wonderful talk. And we're going to have lunch in the Ligonaya, and then so there will be somebody guiding us just outside, and then we reconvene in the early afternoon, sharp with the timing.